We have two presentations that we're about to get going on. Our first is a virtual presentation that will be coming in from Alanna Fulkerson from Texas A&M University. Alanna is going to be talking with us about fish surveys of the Mexican side of the Santa Cruz River and the neighboring Rio Concepcion with environmental DNA. So this is an update for folks who attended in previous years. Alanna has presented, and this will be a great update on that work. So as we're pulling that up, I just want to thank everybody for being mindful about the lunch. Fortunately, soft tacos are probably the quietest food that exists. So if you want to get some more during the presentations, please feel welcome to. All right, are we good to go? I can talk about tacos more if I have to.
Okay, is my
Taqueria
screen showing?
Juanitos. That's the spot.
Okay.
Yep, oh, there you are. We got you. Thanks, Alanna. Thanks for being here. The floor is yours.
All right. Thank you for allowing me to present again. So I won't say the title again, but I will just get started here. Trying to advance my slide. Okay. So a bit of background review. Our target species was the Rio Concepcion Topminnow, Poeciliopsis jacksulci, which was recently described in 2019. And this species is morphologically indistinguishable from closely related species that it co-occurs with, which include the Gila Topminnow and the asexual hybrid Poeciliopsis monica occidentalis. So compared to historically, a lot of the habitats in its known range are now dry. And these two factors, one, it being hard to identify and hard to find sometimes, is the reason we did an eDNA method to survey for this. Our three main objectives were to survey for our target species, the Rio Concepcion Topminnow, in its historic known range and nearby localities, and then also to determine whether asexual monarch occidentalis was present in the Mexican side of the Santa Cruz River. We were also interested in occidentalis, I should mention that. And our third objective was to determine what other native and invasive species were present in the localities that were surveyed. We sampled 8 to 12 sites, depending on the year. And the yellow portion here that is marked just represents the known historical range of our target species. So we sampled sites both within and outside that range. So along the Alisos branch and the Cospira branch. The reason we didn't sample sites in the middle of the range here is because those sites were dried. So they were unable to be sampled. And we also sampled the Santa Cruz River on the Mexican side near the border, near Nogales. So that is the red dot there. This is a figure showing the relative abundance of features that we recovered between collection years, and this is showing them as a percentage within each sample. And the blue sites here are marking the sites representing the known range of our target species. So within those sites, we recovered quite a few features assigned to native species, such as the Sonora chub, Gila, Topminnow, as well as the asexual. And those were pretty abundant for the Emerus cocospora sites that we sampled. But for some of the sites, we also recovered quite a few invasives, such as tilapia, green sunfish, as well as largemouth bass in some of the sites. The site that I want to expand on a bit for this update is the Santa Cruz site Mascarenas, which is this bottom bar at the bottom of the figure. And for this site, we recovered quite a few features assigned to longfin dace, Gila, Topminnow, and the asexual hybrid. But surprisingly, we also detected some features that were assigned to a sucker and some other chub. We weren't sure what those features were representing. And they don't really show up on this figure just because they were at very low abundance compared to these other natives that we recovered. So previously in that area, the desert sucker and Sonora sucker have been reported, but unfortunately, neither one of those species had sequences for the mefish region publicly available. So that was the region that we targeted. But thanks to Jessica Rick, she donated 3 sucker samples that we were able to sequence the mefish site. So there were 2 Clarkii specimens, one from Aravaipa, one from Sonoita Creek, and then there was a insignis specimen from Aravaipa. And those are the ones that we were able to sequence. Another species that is known to occur in that area is Gila intermedia. And there are some sequences for this species publicly available, but not specifically for that site that we surveyed, which was Mascarenas. So first I'm going to talk a bit about the Catastoma story, and then the next slide I'll talk a bit about Gila. So this tree is just showing the sucker features that we compared. So the red is representing our features that we recovered. We had a little over 25,000 sucker features that we recovered. The blue is designating the samples donated by Jessica Rick that we were able to compare to. So our samples most closely grouped with insignis from Aravaipa West, but they weren't an identical match. They were also closely related to the razorback sucker and flannelmouth sucker, which are known close relatives of the Sonora sucker, or C. insignis. And our features were quite a bit different from those Clarkii specimens, as can be seen in the tree. For the Gila sequences that we recovered in that site, we recovered around 4,000 features. We weren't sure what Gila it was. And those features were specifically at that site. They weren't recovered at any of the other sites that were surveyed. The closest match when we ran the blast for those features were to Gila species 1460 from an unknown locality. And they were 1 base different from that species. When we examined it compared to other Gilas, we found that our features grouped with the Gila robusta complex, but it is still quite a bit divergent. It was around 6 bases different from that group. I do want to mention that the features that we recovered at that site do not group with Detainea, it's at least 5 bases different from Detainea. So we concluded that we detected a member of Gila robusta species complex at that site, but it is quite divergent from the other sequences that we were able to compare to. So in conclusion, we didn't find any features that 100% matched our target species reference, the Rio Concepcion Topminnow. But we did recover features in the Santa Cruz that matched the asexual Monica occidentalis and occidentalis. We found evidence of 2 other fish taxa that we weren't expecting at that site, the sucker and the Gila. And so the sucker is likely Catastomus insignis or the Sonora sucker, but it is divergent. And then the chub is likely a member of the Gila robusta species complex. We did detect Detainea, but right now we're suspecting that is a contaminant because some of our other sites did have Detainea, and that are in the known range of Detainea. So we, like I said, we expect that is a contaminant. For the most part, our results agreed with the species that were observed at the time of sample collection. And unfortunately, we did recover quite a few features that represented invasive species, and they were present in every site that we surveyed. And with that, I will take any questions. Thanks for the question. I have read some Hedrick papers. I'm not sure that I understand this question about methodology detecting hybrids. Are you referring to visual surveys or some other type of surveys? Like eDNA. Okay. I'm not familiar with any specific Hedrick surveys that use DNA. If so, could you repeat the last part that I could what? Oh, I think we searched for Gila intermedia, but I think there may have been the issue where the region that was publicly available wasn't a region like it wasn't the me fish region. So we only examined the me fish region in the study. We didn't look at any other region. And if some of the sequences that were available did not have that region, then obviously we couldn't compare to those. Thank you.
River in Arizona. The floor is yours.
All right, so let's start talking about everyone's favorite endangered fish, Gila Topminnow. And it was endangered. It was pretty limited for a long time. Its stronghold has always been the Santa Cruz River for as long as it's been endangered. But we'll just skip through most of this because there's more fun later on. And essentially, this is just explaining what we're calling Upper and Lower Santa Cruz. You've been here all day. You know the Upper Santa Cruz is south of us. Lower Santa Cruz is right here in town. That's the way I've been organizing my most recent data, so we're going to keep moving. And Topminnow had been gone from the Santa Cruz River proper for about a decade, and then they reappeared again in 2015. It was a really big deal. Doug Duncan and Ross Timmons were out there surveying for fish, and they found Topminnow in the Santa Cruz in the Nogales Reach. And that was after a recent upgrade of a wastewater treatment plant, the International Wastewater Treatment Plant. And it was a pretty big deal, you know, endangered fish popping up in the Santa Cruz. But there's more to the story than that. And very quickly after detecting Topminnow for the first time, we expanded into what we now know as the annual fish surveys. Claire Zugmeyer from the Snort Institute has done a great job of leading that effort. And the first issue we ran into was field ID of Topminnow and another very closely related fish was really difficult. So we started collecting both museum quality specimens to go back and process the lab and then genetic specimens. And so the original questions that we had to face were what poecilids are in the Santa Cruz, which we'll get to that in a minute. And then what are their abundances distributions like? How does this change over time because it's a dynamic system? And most importantly, can we morphologically ID all these poecilids in the river? So the two big ones that we knew about going into this was the western mosquito fish, Gambusia. We're going to refer to them as GAAF in the presentation. Then we have Poeciliopsis species, which includes both the Topminnow and the hybridogenic species that we're going to dig into later. Generally, Gambusia are the biggest threat to Topminnow anywhere they're found. Until the Santa Cruz River case, there were 0 examples of Topminnow coexisting with Gambusia for more than a year. And that was one of the reasons why we ramped up these surveys so quickly, trying to figure out how is this coexistence going on. So that was the last time you'd see a whole fish, by the way. Things are very quickly going to get very technical. So here we have a comparison of both of the species. If you look, all fish have 2 sets of teeth. They have oral teeth, which is the same ones that we have in the front of the mouth. And they have pharyngeal teeth on the gill arches in the back. Essentially, there's some fun stuff that goes on with that in diversification, but we're not going to get into that right now. If you look at Gambusia oral teeth, they're these small, spiky teeth, well spaced on the upper jaw. Topminnow oral teeth, they're these big, raker, spatulate things. And so that doesn't tell us much initially, but then when we took the fish apart and looked at their pharyngeal teeth, you can see some big differences between the 2 species. Gambusia have very poorly organized pharyngeal teeth. They're just spikes. It looks like they're mainly meant to hold prey in their mouth and force it down their throat. Much more predatory looking set of pharyngeal teeth. While Topminnow have more of these organized, fine raker type pharyngeal teeth. And so generally it looks like Gambusia will function more in a limited predatory role as a fish, while Topminnow can eat a lot more and have a much broader diet. And that's supported actually by their skull morphology. So Gambusia, if you take a look at these bones right here, this is a premaxilla. Fish have much more interesting bones than people and mammals do. They have a separate moving bone at the front of the jaw called a premaxilla. that attaches to a set of other bones that eventually attaches to the skull. On a Gambusia, they have this really high, really large process, and that means that their jaws are essentially incapable of shifting position and twisting around. While on Topminnow, you don't see that bone popping up. So that essentially means that Topminnow have a much reduced ascending process of the premaxilla. And what it means is that Topminnow are able to flip their jaw up. Gambusia can only pick at food. Topminnow can flip their jaw up and more efficiently scrape food off of a surface than a Gambusia can. And you know, this is, so first year we detected Topminnow in Santa Cruz. This is just Topminnow Gambusia comparisons. First year, it was all Gambusia 2015. We only found a handful of Topminnow. 2016, Topminnow were there. They weren't abundant, but they were there. And very rapidly after that, Topminnow actually became the dominant fish species in the Santa Cruz River. Which, if you heard my earlier comment about Topminnow and Gambusia not coexisting, that was shocking to most people. And then if you pop down to the Tucson stretch of the Santa Cruz, same story. Topminnow are generally more abundant than Gambusia. There's some interesting stuff that we might get into later in 2023, 2024. But as a rule, there's more Topminnow than Gambusia in most sites in the Santa Cruz. So when we were getting all these Topminnow, the first question that Peter happened to have was, why are we getting so many female fish? If you look right here, you know, typically most fish, you'll have a pretty even sex ratio. In live bearers, sometimes it's flipped more female than male. But it's really rare that you get no males at all in your collections. Or you'll get maybe 3 or 4 males. So going into that was when we had these questions. That's when the DNA samples came in really useful. Because we found out that the hybridogenic species, Poeciliopsis monaca occidentalis, was in the Santa Cruz. There had been previous work done in the Santa Cruz that found that that species wasn't in the Gila River Basin. If you want to look at my beautiful map of Sonoran rivers right here, there's 2 fish involved in this. It's Poeciliopsis monaca, the headwater livebearer, which only exists in the Rio Mayo south. And then there's Poeciliopsis occidentalis, our lovely Gila Topminnow, which only exists in the Rio Mayo north. When there is this habitat overlap, because Topminnow aren't very picky, they'll hybridize really readily. And so the hybrids have been documented both in the range of overlap in the Rio Mayo, all the way up into the Rio Concepcion, which is right across the border in Ambos Nogales. We'd known the hybrids were there. They'd been documented really well. And when I say hybridogenic, they don't do what you'd normally think they would do. A female hybridogenic species, the hybrids, will breed with a male of the pure species. What she does when she's producing eggs, she kicks out the old male genome that she was carrying, copies over her monaca portion of the genome, mixes that with the new male genome from the male she breeds with, and essentially produces a first-generation hybrid again the next time they breed. And so that's really one of the reasons that most people think that these hybrids have been able to spread so far out of the range of overlap, because they're able to integrate into the Topminnow populations that they're invading. And so our big questions were, what's going on with these hybrids? How do they coexist with normal Topminnow? What's different about the morphology? And so the big trait we're looking for, which was discovered back in the 60s, was there's a tooth patch on the hybrids that isn't present on pure Topminnow. Topminnow have typical oral teeth. You remember the oral teeth from earlier. The hybrids have the oral teeth and this dense patch behind them of these tiny tricuspid teeth. And so when you take into account that Topminnow are better adapted for scraping, this probably means the hybrids are much more efficient in their feeding behavior than pure Topminnow. And what's probably going on is these hybrids need, most important aspect to remember this slide is they have different teeth. They look kind of different, big things of teeth though. And the hybrids need the Topminnow to coexist. The hybrids cannot exist without a parent population of Topminnow. Let's get into the fun part. What does the data actually look like from the Santa Cruz? So this is here in town, right down the road. Orange is the hybrids. We have not detected many pure Topminnow at all in the Santa Cruz River in Tucson. So Roger Road, initially the first time we sampled there were a few pure Topminnow. And then since then, we have found none. The only site that has had an increase in Topminnow is the Heritage Reach, where we went from detecting no pure Topminnow in the first couple surveys we did to finding pure Topminnow pretty regularly. And if you want to look, this little gray line within the blue line of the data, that's how many of those Topminnow we found were males. And so if you look at Heritage Reach, we're actually getting both male and female Topminnow popping up. So that's a sign of a pretty healthy, integrated, hybrid, pure individual population. Silverbell might be doing something, and Roger may be doing something weird. We'll have to, I'll mention that one a little bit later. And Cortero, when we found Topminnow there, it was just the hybrids. This is a much more interesting story. This is the Upper Santa Cruz. So what we got, this is the farthest south site we've been able to sample. This is Hudson Ranch. We only sampled it in 2024. It's got a pretty good proportion of Topminnow to hybrids to males. And then here are more of our long-term sampling sites. Chavez sighting, whenever we find Topminnow there, it's all the hybrid. It's all just dispersal from upstream. I think we found one male there in 2022, and that was our Topminnow. Potrero Creek was sitting pretty steady with a good Topminnow to hybrid ratio for a while. But in 2024, we struggled to find any fish there. And so all the fish we found were hybrids, all 3 of them. Rio Rico is also sitting really even when you look at their hybrid to Topminnow ratio. The amount of males we catch varies year to year, but there's usually always Topminnow at that site. And Santa Gertrudis had a really high Topminnow to hybrid ratio. And then lately, it has been dropping very fast. And then Tubac, when we were still finding Topminnow there, it dropped really low, very small proportion of Topminnow, and then it blinked out in 2023. We'll have to see what happens in 2025 when we go back and resurvey. Topminnow might pop up again. They are known for occasionally disappearing and popping up somewhere else. But what does this all mean for the future of these Topminnow and the hybrids in the Santa Cruz? Well, what's probably going on is that the only reason Topminnow are surviving in the Santa Cruz is because these hybrids are abundant enough, they're there on the landscape, and the hybrids are good enough at separating their niche from feeding from Gambusia to a different food source, because they have that tooth plate that allows them to scrape better. And because the hybrids need to have Topminnow around to survive, Topminnow are being dragged along by the hybrid populations that can compete with Gambusia. And of course, now the question is, what else is going on in the river? If you've read the Living River reports, you know that there's other fish in the Santa Cruz alongside these Topminnow and their hybrids. One of the big questions we have, especially with the lower Santa Cruz sites, is are other fish affecting the Pomo to Topminnow ratio? And this could be due to predation. Male Topminnow are generally very flashy. They also don't care much about predators. They turn black and they only focus on spawning with females. And so in the lower reach of the Santa Cruz, there's a lot more introduced fish in higher abundance that could be kind of pushing our ratios away from Topminnow. And the habitat is kind of different, but there's a lot of moving factors that could go into this. And then how does flow affect the Topminnow to hybrid ratios is another question we have that we haven't been able to dig into much at all. And so we're going to keep looking at specimens because there's whenever you think you answered something, there's always 5 more questions that pop up. Here we have another nice microscope picture, but this is from a Topminnow collected in 1943 from the Santa Cruz River in the Low-Keel area in the San Rafael Valley. And it has secondary teeth, similar to the way the hybrids do. But the only difference I've been able to find so far is that the tooth patch on this fish was not well done, was not well developed compared to the hybrids, and it was spatulate teeth compared to the hybrid's tricuspid teeth. And then I've also recently found modern Topminnow that have the same tooth formation going on. And we're actually going to try CT scanning all of these fish at some point soon so we can get nice models of their skull. And we're doing head and body morphometric measurements to try and see if there's not tooth differences we can pick out. Then we're going to go to add in some more data from sites with only Gambusia. We're going to try and look at that, look at a few more samples we have. And yep, in 2017, all this research was started. It got kicked off by Peter saying, oh, I wonder why there's so many female fish in these samples. And yep, thanks for all my funders. Thanks for the Snow Institute for running the fish surveys. And we have time for questions.
Let's kick it off on Zoom. I'll do a walking question of my own. So some of the locations like Cortero, right? We saw the spike of the presence of the hybrids, and then it crashed out. Could the population crash out be due to an absence of any breeding partners in the pure occidentalis? Yeah. Yes. Zoom questions.
All right, we have a lot of Zoom questions. From Ken Kingsley, is there any way to distinguish species versus hybrids in the field with live fish?
I was going to attempt to, but holding the fish's mouth open and looking at its teeth while it's still alive and kicking is difficult at best.
Kimmy, I still arrived a little bit late. Apologies. Can someone clarify what exactly the hybrid is? Gila Topminnow and the Topminnow from Rio Mayo, or?
Yep, it's a hybrid between the Gila Topminnow and a Topminnow species from the Rio Mayo.
Mariana Mateos asks, were all your results based on teeth morphology? How confident are you in using the teeth morphology to accurately distinguish them?
I'm actually very confident on the tooth morphology. That was how it has all historically been done with Topminnow. Even, I'm trying to remember the guy's name who originally did it. Big fish guy. He's the one who actually identified the hybrids. That's the way Jack Schultz did it when he was identifying all these hybrids. And it's a pretty distinct difference between the two. If you want to get really specific, Topminnow consistently have those, if they have secondary teeth, they're spatulate, while the hybrids seem to be pretty consistently tricuspid. And we did compare 2 genotyped specimens, and we found the same pattern holds true.
So it also seems like the success of the Heritage Project is paying dividends again and again and again, right? We're seeing increase in the occidentalis there as well. To what would you attribute that? Is it hydrologic connectivity? Is it the fact that those were introduced from a pure source? All of the above? Well,
the source for the Heritage Reach Topminnow is the upper Santa Cruz, so they were mixed from the beginning. But it's likely due to they've always had occidentalis in the hybrids. It's just recently the abundance of occidentalis has gone up. I'm going to attribute that. We won't know enough. We don't really know enough for me to say for sure. But I'm personally going to attribute it to a lack of invasive fish species and a relatively stable hydrologic regime. But right now, we don't know enough about those relationships to say anything for certain.
Alanna asks, you mentioned contemporary samples. Have you been able to examine any historical samples?
I've only examined the 1943 samples. If anyone has friends at ASU that can get us into the fish collection, that would be great. They have many more historic samples of Topminnow that we can look at.
Does anyone have friends at ASU? All right, there we go. We got a hand in the back of the room. All right, Matthew, great job. Thank you so much. So this brings us up to our first video of the afternoon. These videos have just been so good. This is Meet the Turtles of the Santa Cruz River.
Running through and northwest of downtown Tucson, Arizona, lies the beautiful Santa Cruz River. This stream follows a long course from where it starts near the US-Mexico border before traveling over 150 miles to where it meets the Gila River near Phoenix. In modern times, the Santa Cruz River is what is known as an effluent-dependent stream, meaning the water in the river is supplied by wastewater treatment facilities, and it depends on us humans to keep water flowing in the river year-round. Many different plant and animal species call the Santa Cruz River home, but some of the real stars of the show are the turtles. During trapping surveys conducted by Professor Michael Bogan and students from the University of Arizona, along with local K-12 teachers in Tucson, 3 species of turtles were found to live in the Santa Cruz River, and they couldn't be more different from each other. First, there's the Sonoran mud turtle. This adorable species is native to the Santa Cruz River and has called the desert Southwest home for a long time, likely at least 1 million years. Mud turtles are the smallest of the species found in the Santa Cruz. They usually weigh around 7 to 11 ounces, that's less than a pound, and have a shell that's no longer than 6 or 7 inches. They spend most of their time in the water, buried in sand or mud, or hunting around for a meal. Mud turtles mainly eat aquatic insects, small fish, or snails, but they can also eat small aquatic plants, algae, or even mesquite pods from trees around the river when competition is high or food availability is low. If you go out to the river looking for mud turtles, you probably won't see any. These little guys love to hide, and their population is only a fraction of what it used to be due to habitat loss and the introduction of non-native species. The second species of turtle found in the Santa Cruz River is the spiny softshell turtle. This is an invasive species, and unlike the native mud turtles with their hard, protective shells, spiny softshell turtles have a much weaker shell that almost feels like leather. These turtles are aggressive predators that will eat just about anything they can catch, including fish, frogs, and insects. This species is the largest turtle that lives in the Santa Cruz and can grow to over 1 foot long and weigh up to 7 pounds. Spiny softshells also look different depending on if they're male or female. The females are big and tend to be feisty, while the males are smaller and have distinctive pink marks on their head. The third turtle species that is common in the Santa Cruz River is the red-eared slider. And just like the spiny softshell, this species is also invasive. The red-eared slider's most distinctive trait is the red stripes on the ears of all individuals. Because red-eared sliders are so cute, they are one of the most popular pet turtles in the United States and across the world, and as a result, they're found on nearly every continent. If you were listening closely, you may have noticed that 2 out of the 3 types of turtles found in the Santa Cruz River are invasive species. But what does that mean exactly? An invasive species is a species that is not native to the area it currently lives in. These are species that have likely been introduced to an area in the recent past. This can happen in multiple ways and for many different reasons, but here in the Santa Cruz River, the invasive turtle species, the spiny softshell and the pond slider, likely arrived by people releasing unwanted pet turtles into the river. This is why you should never release your pets into the wild under any circumstances. Invasive species cause huge problems for both their new habitat and the native species that have evolved millions of years without them, like the Sonoran mud turtle here in the Santa Cruz. Invasive species compete directly with native ones for often scarce resources, and can also prey on vulnerable native species who aren't used to them being there. The two invasive turtle species, the spiny softshell and the pond slider, have made life for our native Sonoran mud turtle extremely difficult. There are not a lot of mud turtles in the river now, but before the introduction of these invasive species and before urban growth greatly altered the river's habitat, the Santa Cruz supported a much larger population of mud turtles. Restoring the population of a native species is not easy, and the population of mud turtles may never return to its historically large population numbers. But people like Michael Bogan and his lab at the University of Arizona, who really care about Arizona native species and the mud turtle in particular, are working hard every day to help start the process. If you are interested in learning more about the Santa Cruz River and all the species that live there, visit santacruz.arizona.edu.
All right, thank you for your promptness. Okay, so we have a block of 3 presentations right now that will conclude with a video. We're going to be hearing about terrestrial insects, or are they? We're going to be hearing about some aquatic invertebrates, and then about some local drought response coordination frameworks. And to start this all off, I'd like to welcome first-timer Meredith Willmott from the University of Arizona, Hopping on the Santa Cruz, Novel Aquatic Adaptations in a Terrestrial Insect Group. Welcome. Kind of stay right around here so you're in frame, okay?
Right here? Okay, and then this is...
That's just easy, front and back. The pointer doesn't work. Just front and back.
All right, thank you. Thanks for the introduction, and hi everyone. I am so thrilled to be here today to tell you about a member of the Santa Cruz River insect community that many people may not have noticed before. So it's a natural history story of epic proportion, I think. So I am a first-year PhD student with the Department of Ecology and Evolutionary Biology here at University of Arizona. So I'm going to provide a little bit of context for why I think this observation is so incredibly fascinating, tell you a little bit about the organism that I'm working on, my system, go over the big questions that we still have to answer from this, and then what's the future? What's happening next? So, especially here in the Sonoran Desert, with the amazing biodiversity and abundance of all forms of life, you go out into your front garden, you see this incredible diversity of plants. And the Sonoran Desert is actually a world biodiversity hotspot for a group of insects called the Occidentalis, a group containing cicadas, leafhoppers, plant hoppers, and tree hoppers. The reason for that is because these insects are plant feeders, sorry, I have a little bit of an accent. They're plant feeders, and they have diversified alongside their host plants. So this diversity of plants has resulted in a huge diversity of form and function. And there are 3 major hypotheses around why. So we think that maybe insects have low extinction rates. They have a resilience to extinction. They have acquired new adaptations and new forms and new functions that have allowed them to spread into these different niches. And then the niches have appeared and the insects have moved into them. And so this is really about number 2. So insect evolution, just for those of us who don't spend all of our time looking at insects all day, is characterized and facilitated by really high reproduction rates and their rapid ability to evolve in response to novel environmental pressures. So now I get to talk to you about hoppers. So like I mentioned, the Sonoran Desert is a global biodiversity hotspot for all these different groups of hoppers. These are photos by an absolutely phenomenal local macro photographer, Salvador Vitanza. We have this little pink plant hopper. We have tree hoppers, a tree hopper nymph, leaf hoppers in the corners here. And this is the leafhopper that I'm going to be focusing on today, Draculacephala californica, the false-grass sharpshooter. And with their amazing abundance, their huge diversity, and how common they are, you might be thinking, Meredith, I've never seen one of these. And that's because here's an accurate depiction of their size. So to scale, here's a leafhopper picture on the screen. Most of these are going to be around the size of a head of a pin. They range from 3 to 25 millimeters in length, and they hop. They move at the speed of light. So good for the leafhoppers that most people can't find them. So they are globally distributed. They're fully phytophagous. They're fully plant feeders. And their diversity is, again, strongly associated with that of their host plants. They're also fully terrestrial, which makes us a weird insect group to talk about the Santa Cruz River meeting, right? You would think. So Draculacephala californica, the false-grass sharpshooter, is an endemic species to this region. It's phytophagous and terrestrial, but it's also known to be hurricane dispersed, where it becomes a pest in Florida. It's picked up by hurricanes and moved around, and it's associated with aquatic plants. There are many leafhoppers and plant hoppers and tree hoppers that are associated with aquatic plants, but we still think of this group as terrestrial, despite observations on citizen science platforms such as iNaturalist documenting this insect on the surface of the water. Now, the issue with documenting an insect on the surface of the water is we don't know if it's alive or dead or moving around with any sort of efficiency, right? Which brings me to my novel natural history observation. In the fall of 2024, Scully, our next presenter, brought me out to this amazing site along the Santa Cruz River, and I saw this leafhopper moving around on the surface of the water. It's associated with this water primrose here, Ludwigia, which is a secondarily aquatic plant, meaning that it evolved from the water onto the land and then moved back from the land into the water. Don't ask me anything else about that. I'm not a botanist. But one of my big questions here is, when that happens and you have an insect that's feeding on a terrestrial plant, the plant decides to go back into the water, what happens to the insect? What does it do? In the case of Draculacephala californica, I think you need to play for me, I'm sorry. A little bit of a teaser trailer here for you. Oh no. Well, it's swimming. I hope that I can show you, thank you. It is swimming. Well, even though I just told you that it's difficult to tell from static photos whether an insect is moving efficiently across the surface of the water, I suppose you will have to take my professional word for it, that they are swimming. And this gives me an opportunity for one of my least favorite things in the world, interpretive dance. So this leafhopper, we've all seen water boatmen, right? And water skaters, sometimes called Jesus bugs, using their hind and middle legs to propel themselves over the surface of the water in a movement that looks kind of like this. Good God. Thank you, everyone, for holding with me for this. So, not going to do that anymore, but you get my drift, right? So this leafhopper was doing that too, which is crazy because they're not supposed to do that. So why on earth are they doing that? And are they doing this because they're falling into the water and they're struggling and they just really need to get out? Or are they doing this because, well, maybe they've evolved back into the water alongside their host plant. Maybe they are adapted to this aquatic environment and they've been around it for long enough that they've overcome the challenges associated with aquatic living, which are numerous. So, aquatic insects. You'll hear more about this from Scully, so I'm not going to go too in-depth here, but most of the aquatic insects that we really know about in this order of insects, Hemiptera, which is also the order of insects containing my hoppers, is that they're predaceous, they're fully aquatic, and they have legs adapted for swimming and hunting. Not so the hoppers. They are phytophagous, terrestrial, and they have legs that are adapted for jumping and walking. So what are the questions here? What are we doing next? So when I was fresh out of seeing this leafhopper propel itself across the surface of the water like something that isn't a leafhopper, I was immediately caught up with this question of, well, how is surface type affecting this mobility? Are they equally efficient on water and land? So Aquatic insects, they have to overcome a variety of challenges associated with aquatic living, and one of the biggest challenges is surface tension. We've seen insects bogged down in pools, bogged down in streams, and this leafhopper, despite having wings that were occasionally being caught up in the water's surface, were remaining mobile on top of it. So clearly, they're able to escape surface tension. About 40 hours of observation that can speak to that. This is a video showing you a jumping leafhopper moving out of the water. So I went out into the Santa Cruz River, and for a very long time, I knelt down knees deep, and I collected leafhoppers with my hands and an aspirator and took them out of the water and put them into these leafhopper mobility contraptions here, little plastic tubes filled with water and then not filled with water. And I measured how long they were moving horizontally against a meter stick. And I did this with my Draculacephala californica, my false-cross sharpshooter, then with a couple other sharpshooters that were just along the banks on the vegetation, not in the water, so being normal leafhoppers. What I found is there's absolutely no significant effect of surface type on the leafhopper's distance jumped, which to me indicates pretty clearly from this pilot data that they have found a way to successfully adapt to that major challenge associated with aquatic living. So future research I'll talk about in a little bit focuses on understanding what and why that adaptation is around. And I will say, for the record, in my tests with comparing this species jumping on solid and fluid surfaces to other species, the other species died. This was a very high mortality rate leafhopper project, which was sad for me, but added more compelling evidence to the idea that this leafhopper, associated with this secondarily aquatic plant, might be moving back into the water itself. So what's the future? What am I doing next with my very strange aquatic leafhopper? So how has it moved into aquatic environments, and are there other species displaying similar aquatic adaptations within the region? So this summer, I'm going to be going up and down as many sites along the Santa Cruz River as I can to identify other species displaying this behavior, and I'm going to be taking them into the lab and observing a few different morphological characteristics that I think could be contributing to this success. Thank you. So one of these is body color. They are camouflaged in a way that protects them from predation from below and predation from above. This is how leafhoppers are typically colored, so could just be a pre-adaptation, that's something that's helping them out. Could be their leg lengths. They have very, very long hind legs, and these legs could be distributing their body weight evenly enough across the surface of the water that surface tension they don't even need to respond to these effects of surface tension because they're not breaking it. Then, perhaps most interesting are brochosomes. Leafhoppers are the only insect group that secretes this tiny soccer ball-shaped particle, and it's hydrophobic, and it's produced continuously throughout their life cycle. So the leafhopper, in another video that is at the end that I will not play, has this very characteristic motion of taking its long hind legs, reaching down to its anal opening where the brochosomes are produced, collecting those brochosomes, and then smearing them across its body, using the spines on their hind legs to assist with that distribution. So this observation, to me, has enormous implications for the radiation of plant-feeding insects into aquatic habitats, and really challenges our pre-existing assumptions of what herbivores do when their host plant moves back into the water, which is a question as of yet unexplored from the perspective of insect diversification. Also, highly unlikely that this is unique, which is why I'm so excited to do more research this summer. This is just a very brief overview of everything I've just talked about. I'm very, very excited to go on with my research this summer. And if you are interested in learning more about this, you can email me below. I'm happy to share videos, and they are also on my website, which is that QR code if anyone would like to see the swimming leafhoppers.
All right, we certainly have time for questions. Yes.
Where do they lay their eggs, and what do they do during their larval stage, and is it any different than the other leafhoppers of the region?
Great question. So they do lay their eggs on their host plants, but they are generalists. And I was a little bit late into their generalists, just meaning they'll use a bunch of plants instead of just one specific plant. Because I was seeing them so late into their breeding season last year, I don't know if they're laying their eggs on that Ludwigia, that semi-aquatic plant, but it's definitely something I'm going to try to monitor or at least find out this summer. As far as their life stage goes, I did actually collect nymphs that were displaying this same aquatic behavior, which was really interesting to me. Ooh, no, really quick though, the eggs are covered in brochosomes. They are covered in that hydrophobic particle. So we know that's true across leafhopper species. It serves to protect them from getting trapped in sticky sap or their very sticky poop.
Does the parent do that, or do they come out that way?
Come out that way, out the same tube.
Oh, same tube. The B tube. All right, other questions? Wow. Well, thank you.
Yeah.
Truly, that was very impressive. You can stick around for a little bit if people have more questions. All right, Meredith, great job. Thank you. All right, so, oh yeah, okay, great, Scully. So next up is Scully Young. Scully's is here from the University of Arizona, and we'll be talking about impacts of drying and flooding on aquatic invertebrates in 4 reaches of the Santa Cruz River. Scully, the floor is yours.
All right. We'll take a moment. I can introduce myself. My name is Scully Young. I am an undergraduate research technician at the University of Arizona, working with Michael Bogan in the Department of Natural Resources. And I'm going to be talking to you guys about the aquatic invertebrates that are living in the river. So thinking about our damselflies, dragonflies, snails, mayflies. For sure. Yeah. That's my spiel at the beginning. I have like pictures to talk about.
Are we sure we don't have any questions from the previous presentation? Okay, yeah, Matthew's got one in the back. I got you.
Maybe we can tag team this.
You guys can do this.
Can be the aquatic invert team here.
Is leafhopper diversity, it's a 2-part question.
Awesome.
Is leafhopper diversity generally associated with higher plant diversity?
Yes.
And do you
100%.
think if you go to spots with a higher diversity of aquatic and riparian plant species, you'll find more leafhopper species?
Great question, one of mine. I strongly believe that, yes, I completely think that that's what's happening. So that's one of the reasons I want to know how many species are displaying this behavior. And there were pictures on my last slide there of leafhoppers in completely different genera than the group that I'm looking at that have been observed on these citizen science platforms as on top of the water associated with aquatic plants. There are plant hoppers that nest only at the base of aquatic plants. So there's lots out there to uncover.
So also something I noticed, I know within the box and whisker plots of your pilot study, right? While the overlap was suggestive of equal efficiency in sort of dry or on surface, the average jump length seemed to be longer on water. Did I see this correctly, and am I overinterpreting anything?
No, you saw that absolutely correctly. One of the things that I was sort of curious about when I was starting that is that I thought maybe it had to do something with just the water motion itself, and that there was like an imperceptible difference that I wasn't picking up on from placing the leafhopper on the water with my tweezers and watching it jump. So that's what I think accounts for that so far.
All right, thank you. And we were 5 minutes ahead of schedule, so thank you, Scully, for everything that you've done so far to put us back on schedule. Everybody. All right, all yours.
Good. All right. So Something important to understand about the Santa Cruz River, and especially the flow in the river, is that it's very dynamic, right? So we can see from these 3 pictures here looking at the Heritage Project in downtown Tucson. This first image on the left is showing when there's the normal flow coming out of the outfall pipe at Heritage, but then there are also periods of time like during the sediment removal where the reach was completely dry. And there's also the seasonal monsoons that completely flood the channel with a lot more water than there normally is. So what I'm going to be going over is getting a look at how the aquatic invertebrates are responding to these very dramatic changes in the flow in the river. So a brief history of what's going on in the Santa Cruz here in Tucson. We had historically perennial reaches that were groundwater fed, but those all dried up in the early 1900s due to excessive groundwater pumping. And so since the 1950s, all base flow has been treated wastewater. Initially, that wastewater was pretty poorly treated, and there was very little aquatic life in the river, but modern technologies and upgrades to those treatment processes that we heard about this morning increased the biodiversity. Now there's a ton of aquatic life in the river, but we have multiple sites here in Tucson that are releasing effluent, and they have varying histories and current management that we'll get a little bit into. And we also do still have seasonal natural floods from the summer and winter rains. So getting a look at, on a map, what our effluent release sites are here in Tucson. We have 4. These 2 purple dots on the map are the original effluent releases that have been flowing for more than 50 years, with those upgraded, really high quality water coming through since 2013. Then moving upstream a little further south, we have the Heritage Project, started in 2019, and even further south, our newest site is the TARP plant releases, started in November of 2021. So taking a look at where our sites for this research fall along this map, we have Tangerine, which is pretty much our reference site because it's in that really long flowing area of the river. That's way out in Marana. Then we have Cushing, which doesn't have its own outfall pipe, but is instead downstream of the Heritage Project, which is at Star Pass. And then we have our newest site, Irvington, which is by those TARP plant releases. So getting a look at what the flow coming through each of these sites actually looks like throughout our study period. So going from January 2021 through the end of 2022, we have Tangerine here, which has really high base flow. And we're seeing these big peaks in flow during the summer monsoons, and then a smaller peak during the winter rains. So these are those natural seasonal flooding events that we're seeing in all of the stretches of the river. So we're also seeing those in Star Pass, but the main thing to note here is that the Star Pass base flow is significantly lower. There's a lot less water at Star Pass than there is at Tangerine. Then going downstream of Star Pass, we have Cushing. So we're seeing that when there is base flow at Cushing, it's lower than Star Pass. And we're also seeing that Cushing has these periods where it is completely dry. So that is because during the 2021 monsoons, a bunch of sediment was washed down and deposited here at Cushing. And so the infiltration rate just became way too high. No longer the flow coming from Star Pass was able to have there be surface flow at Cushing. And then our newest site, Irvington, is all over the place in terms of the flow. So water here is turned off and on, several different periods of drying that are shorter, and we are still seeing effects from the flooding, but overall just a lot less consistent in the flow. So getting a sense of what that actually looks like on the ground, we're seeing at Tangerine, consistent high base flow, very uniform channel, lots of water, big, tall riparian plants. At Star Pass, we have that consistent low base flow, lots of riparian plants, loving that consistency, but just a lot less water. And then looking at Cushing down the road, characterized by its long drying periods, lot less flow, less aquatic plant life. And then we have Irvington, newest site, water's turned on and off with those recurring drying periods and started much more recently. So the main questions that this research is getting at are, one, how do these changes in flow regimes, so those flooding and drying events, how are they affecting the aquatic invertebrates in the Santa Cruz? Also, which species are standing out by either doing really well or really poorly during these flooding and drying events? And how do flow release management practices affect the aquatic invertebrates here in the river? So we did monthly aquatic invertebrate sampling at all 4 of our sites throughout our study period. We brought those samples back to the lab, sort them out, identify the species, and then from that, we're able to calculate species richness as well as different factors of community composition, and comparing what you guys are gonna see with these graphs is comparing those values between the different sites and also when there is drying and flooding. So this first graph we're looking at here is looking at taxon richness. So this is just going to be the number of different types of bugs we found in a sample. First, we're looking at tangerine, which we can see has a really high species richness throughout our study period. It's also very consistent. We have these 2 main drops in species richness, which align with the monsoon flooding. So floods come in, wash a bunch of species out, but then after a month or so, those species come back to the river. Then we are seeing those same dips when we're looking at Star Pass. Star Pass, however, has a lot wider of a range in species richness than we're seeing at tangerine. One of the reasons that this can get so much higher than tangerine is because there's a lot more variety in like microhabitats at Star Pass. So you're seeing in that picture, there's like little pools and shallower riffles. Different species are gonna prefer that to the like uniform channel at tangerine. But generally this is a lot less stable but high species richness with those same effects from the monsoons. Then looking downstream of Star Pass, we have Cushing. So long periods where it is dry, there are no bugs there because there's no water there. But what we are seeing here is that when floods happen, so this peak being during some rains in the winter, and then this other peak jumping up being the monsoons in 2022, that actually that is bringing species to Cushing. So it's acting as flooding being a way that is dispersing species to this site, which is really cool. We're also seeing that species richness like jumps up really quickly at Cushing. And that's really evident, especially when we compare it to Irvington, which is all over the place. But starting in November 2021, then goes dry. Then there is flow restored, but only about like 5 species are there that first month. Nowhere near the big jump we see at Cushing. It then takes several months, but then we're seeing a lot of species after there's consistent flow for a while. Then floods happen, then it dries up. And then when flow is restored, we're just not seeing that species richness recover as quickly as we are at Cushing. And then another thing you can't see on this graph is that at the end of the year, there were a couple periods where Irvington was completely dry between samples. So they were just short periods of time, but we're thinking that's probably what's keeping this species richness so low, and it's not recovering the way we're seeing at Cushing. So another thing that we can look at to kind of get a sense of what's going on with the bugs at each of these samples is looking at community composition. So this takes into account like how many species are there, but it's also going to look at abundances and, you know, like if we have 2 samples with 5 species and one of the samples there's 100 of one species and only like 2 or 3 individuals of all the others, or in the other sample there's 20 individuals of all 5 species. Those are different communities. So communities that are more similar are going to be closer together on this plot. So each point is a sample and they're distance is based on if they're more similar or not. So what we're seeing mainly here is that Tangerine has a very consistent aquatic invertebrate community. That's going to be our reference site. It's going, generally, we're just seeing that all the samples look really similar to each other, especially when comparing it to our other 3 sites where it's just a lot more spread out. We're also seeing that Star Pass has several samples that are very close to Tangerine, and Irvington even has a sample where its community is very similar, which is really cool. It's showing that these sites have the potential to reach those really stable communities, but also that all of that flooding and drying and flow stuff is changing these communities a lot between the samples. Another piece of information we can get from this type of analyses is what are our indicator species at each site. So this is basically like what species are unique to this site and very often seen there. So this kind of gets you a sense of like, we're looking at a lot of graphs, but these are the actual bugs we're seeing in a sample. So at Tangerine, we're having some damselfly larvae, some leeches, some freshwater limpets, free-living caddisflies, and amphipods. At Star Pass, we're having dragonfly larvae, predaceous diving beetles, ram's horn snails, and pond snails. At Cushing, we're having this really cool mayfly species that has a really short lifespan, so it does really well in places that dry up, which makes sense, and also some water striders. And then at Irvington, we have some diving beetle larvae that are pretty big, and then we also have some smaller, different species diving beetle adults. So then we were able to look at, for our species, across samples, like how are drying and flooding affecting these species. And what we found is that drying has a pretty strong effect. It either reduces or eliminates these long-lived mayfly, damselfly, and caddisfly species. So these bugs with long lifespans, they're just not doing very well when there is drying. We do, however, have some resistant fly larvae and also winged true bugs that drying has no impact on. So they're either able to recolonize really quickly, or, you know, winged true bugs, they can fly away and then fly back when the water comes back. We're not seeing impacts on these species. And then also something interesting we found is that flooding generally has no impact on any of our species. All species seem to kind of bounce back within a month of floods happening, which is really cool and kind of shows that our bugs here are very well adapted to our seasonal monsoons that we have here. So kind of an overview of all that stuff I just went over. We're seeing that flooding impacts on aquatic invertebrates in the Santa Cruz are very fleeting. We're also seeing that drying is generally reducing species diversity and having a very strong impact, changing community composition, and that only a few true bug and true fly species are going to be resistant to drying. We are, however, seeing that recovery from drying can occur quickly if there is that perennial flow upstream. So when we were looking at Cushing, having Star Pass upstream, the bugs were able to come back pretty quickly, but in isolated reaches like Irvington, we are not seeing that quick recovery. So some management recommendations we came up with based on this information. Keeping base flow consistent increases aquatic invertebrate diversity. The bugs like it when water is there all the time. And even short-term, like less than 24-hour drying events can harm these ecosystems. And then another thing, if there needs to be drying, these multiple outfalls closer together could potentially facilitate ecosystem recovery in the event that drying needs to occur. I would like to thank these people at the U of A and with Tucson Water for helping with this research, and also all the funding for this comes from 3 philanthropic donors. And if you guys want more information, you've heard about the website a million times today, but you should check it out, or you can email me or Michael. Thank you.
Hey, well done. Okay, we'll go directly to Zoom for the first question. Zoom says,
At Zoom, Ken Kingsley says, are there differences in current flow rate and or depth between your sites that might correspond with species distribution?
I'm not quite understand. So current flow, could you say it again?
Current, comma, or like in parentheses, flow rate. So differences in flow rate and or depth between your sites that might correspond with the species distribution.
So we didn't like analyze that specifically, but like anecdotally, those sort of comparing like the flow that we're seeing at each of the sites and the communities we're seeing, like, yes, Tangerine has a distinct community and a distinct like flow rate from the other 3 sites, and it is more stable there. But I think like more analysis could be done to get like specific kind of like relationships with that.
great, thank you.
The sites, the 4 sites, have different substrate conditions, probably. Does that affect the invertebrate community? How different or similar were the substrates that you were looking at?
I would say that like substrate does vary, but it's like relatively similar. There's sand and like bigger rocks and those sorts of things at each of the sites. I would say that like that's not exactly something we looked at either, so I couldn't answer like specifically what's happening with that, but like different bugs do prefer, like some like to bury in the mud and some prefer to be in the sand or some prefer to be under like bigger rocks. So definitely like substrate size can impact species for sure. Oh.
I was just going to add to that to help answer the question for Scully, because we just did these analyses like a week ago, so this is brand new. And we do have substrate data, and it is pretty similar. So it's not like it is below Agua Nueva, where it's like mostly sand. So we don't think substrate is an issue, but we haven't run the analyses yet. But water depth, water velocity, as Ken Kingsley mentioned, definitely could be an issue.
We can get one more. Going once? Yeah, okay. Pardon me, sorry.
Have you explored which species or which groups bounce back first?
That is a good question. We haven't looked at like, at least I don't know like which specific species are bouncing back first beyond the like resistant fly larvae and the water striders. But I do know like something we see like at Tangerine that's really interesting is that there's like a certain mayfly species that seems to do really well when like after flooding it comes in and it's much more prevalent, probably because there's less competition, whereas when the flow is more stable then like Falcion like is like the main species that kind of takes over. So there are definitely certain species that do well in those like disturbed like after effects.
Scully, great job.
Thank you.
All right. So with Meredith and Scully, those are 2 first-time Santa Cruz River Research Days, folks. I think they did a fantastic job. Congratulations and thank you. All right, next up, we're going to bring up Catlow Shipek from Watershed Management Group, and Catlow will be talking to us about developing a local drought response coordination framework. Catlow. Mike's ready.
Great, thanks. You got it. Good afternoon, how's everybody doing? Almost the end of the day, lots of information. I'm gonna dry things up a little bit. We're going to be talking about drought, so hopefully it doesn't put anybody to sleep. But a really important topic, because what are we in the midst of right now? Drought, yeah. So I'm so thankful to be in the room full of riparians. So we love water, we cherish water. So now the question is, during times of drought, what can we do about that from a communications perspective, a management perspective, and a policy perspective to help ensure that these riparian systems are resilient for the long term? Most of you know me through Watershed Management Group. I do wear another hat, so today I'm going to be speaking as kind of my role on the Santa Cruz Watershed Collaborative, that I sit on the coordinating team and the co-lead, and many of you are partners and have participated in either the coordinating team or at recent forums, and or also in the drought planning process that's currently ongoing. So without further ado, just to kind of orient you, this is our area of focus as a watershed collaborative, essentially from the Pima County southern border to the Pima County northern border, and then the areas that drain to the river in those reaches for that reach. So the big question that I alluded to is, how do we find resilience? And a speaker yesterday talked about collective action and the importance of collective action. Being that we are a large urban watershed with many different land managers, landowners, organizations, agencies, jurisdictions, how do we have local water resilience? And not just for us as people, but for our plants, our animals, and our river ecosystems, even now during times of severe drought. So first, making sure that we give acknowledgement to those who have stewarded our land and waters since time immemorial, the O'odham and the Hohokam and the Sobaipuri, as well as now continuing through today, the Pasquayaki and the Tohono O'odham, who continue to steward these lands and waters. And then thinking about, the number of times we've had drought since time immemorial in these areas, and they've continued to steward those rivers through those times. So now how can we pick that up and do that across our basin, continuing today? And just to kind of give you a little bit of mental framework, so we're going to move off the Santa Cruz River for a lot of these shallow groundwater supported systems. So these aren't effluent dominant systems, so they're much more prone to our local or episodic shortages in water supply. This is the Tanque Verde Creek just upstream from Craycroft Road. Our last water year, 2024 water year, this had water for 241 days. No. Yes. I may be misremembering. I'll have to check with Nadira. But this is an intermittent reach. This winter, it's been dry the entire winter. So recognizing wet years versus dry years, average years. But this is part of a shallow groundwater system with cottonwood trees. There are a few remnant walnuts. It probably used to be a cienega system. So a series of wetlands, not even an open channel. Now we have that open channel, but a lot of these riparian trees are dying. And actually, I just went for a run yesterday, and a lot of these cottonwoods have visible water stress. So yellowing of leaves, some dying of limbs. And so they're very much experiencing that water shortage currently. So thinking about this drought planning goal, we had 3 different goals, sub-goals as part of this. Is 1, to develop a watershed-wide coordination of drought planning and drought response. 2 is thinking about how to ensure local water reliability. And 3, ensuring local watershed health, including healthy aquifers and rivers. And as part of the Santa Cruz Watershed Collaborative, the vision of the collaborative is people working together to ensure a healthy urban watershed with flowing rivers and streams. And so this is really cool. This is over, I think our next slide, has 40 different partners aligned on this vision, working on this vision together, each in our own different capacities. But thinking about that collective action, that cooperation, and that coordination that needs to happen. So what is drought and how do we measure it? So there's a number of ways to have drought. There's meteorological drought, there's hydrological drought, there's agricultural drought, there's economic drought. So what we're going to focus a lot on through the collaborative as we go through this planning process is hydrologic drought and then how that impacts ecologic function and impacts. And so we start with looking at rainfall. So with rainfall, we can easily measure departure from normal. So is it better than normal or is it less than normal? And so that can be our early indicator. We know that on a monthly basis. With essentially no lag in time, we can say, yes, we're in a drought, no, we're not, or how severe or how intense it is. We can also have reduced snowpack. So we're seeing a lot of these mountain front creeks and shallow groundwater systems. We have less snow. So that means less snow melt, which means less water feeding these systems. And then more reliant on rainfall, which comes in a flashier response. But we can also have man-made drought. We can have human-caused drought through dropping of aquifer levels, diverting of rivers, even reservoir levels, Lake Mead, et cetera. So recognizing that we can have natural drought, we can have human-caused droughts. So as we started this process last fall, we immediately set up a technical advisory team, and that consists of Tucson Water, Pima County Flood Control District, Pima Association of Governments, Watershed Management Group, and then our SQUAC staff, Maria Johnson, who unfortunately couldn't be here today. And so the first thing we're like, we need a definition. How are we going to define drought here for our local watershed? And then how do we work from that definition? So recognizing, and I'll get to this in a little bit, we're in a long-term drought. So we're recognizing in this definition an episodic deficit. So thinking more of the short-term severe droughts that we're seeing. And a deficit in water availability for our human communities and or our natural communities. So those being the plants and the animals of the lower Santa Cruz watershed caused by less rainfall, less precipitation, reduced stream flow, or a decline in shallow groundwater levels. And that recognizing that drought may be localized, it may be localized to a subwatershed, a stream system, a shallow groundwater system, or it may extend across the entire basin. So, anybody remember 2020? Worst year on record. Some of you have heard the story, right? What happened in 2020? 3 really awful things. Bighorn fire. During the middle of the summer, while we were in the pandemic, so we weren't traveling anywhere, we couldn't cool ourselves off by traveling up the mountain, watching the mountain burn. What was the third thing? Hottest, driest year on record. We were like, why do we live here? And we're like, who's going to do something about it? The city, the county? No, maybe not. And so recognizing why did the city not declare a drought? Why did the county not declare a drought? It was the hottest, driest year on record, right? I was going to the local drought impact group going, how come nobody's declaring a drought? We should do something. And they're all like, no, yeah, we're good. We have water. We have water coming from the Colorado River. So unfortunately, fortunately, our city and county's drought plans and responses are tied to Lake Mead. So how are levels in Lake Mead, and how is that then going to determine our local actions? But Lake Mead is what? Hundreds of miles away. It's local drought. So why are we not responding to local drought? We've so disconnected our water systems, our water supply systems, that we could care less about local drought, except for those who care about our plants and animals and maybe our urban trees. So that got us thinking, what do we do about drought? What can our organization, Watershed Management Group, do about drought? What can Tucson Bird Alliance do about drought? What can the county actually do about drought? So it got us thinking about, we need to think about this differently. And yes, we're in a really long-term drought, over 20 years. So what are we going to do about that? It's a dry topic, I told you. So this is the latest drought status. Not looking good for Arizona and much of the Southwest. And so what's at peril immediately? our creeks and our rivers, and then our shallow groundwater systems. And maybe someday when effluent becomes so valuable, it could be our Santa Cruz River as well. So thinking about all of this demand for water, how do we make sure we have enough water, not just for us, but also our creeks and rivers? So through the technical advisory group, we started thinking about how are we going to define drought? What is going to be the status? And then how are we going to trigger that status to trigger certain actions? And so we were looking back at this. How many of you can think of different actions for each step of that way? And it's a lot of steps, right? It's like, do we need that many steps? And what about when we're not in drought? What are we doing then? And so a lot of people started thinking about, we should celebrate when we're not in drought, right? Because if we only give a doomsday message all the time, it's pretty lame. And there's going to be a lot of apathy. So we need to celebrate those. When the times are good, let's recharge that rain. Let's get that stormwater in the ground, right? And then start maybe simplifying some of these drought stages to then correspond with certain actions and using rainfall as that early indicator. We're also tied in and keyed in to ecological drought, but there may be a lag from when there's a departure from normal rainfall to when that cottonwood starts to show stress. So now we're in our current drought, 2024. I look around the room and I'm like, what are we doing about this? Not much, right? So fortunately, nothing drives drought planning strategy and getting people engaged on this as being in the middle of a drought and experiencing it. So based on this triggering departure from normal rainfall, we've separated a little bit from how the state or the US Drought Monitor says. We've simplified some of these drought stage classes by recognizing last monsoon season, we would have entered early drought stages to then it heightens and intensifies through the winter period because it's been a really poor winter. So that's where we're at. Where are we going from there? We can only hope for a good monsoon season to kick us out. And we're also seeing that these droughts, this is the 2020 drought. We saw a strong correlation with decline in shallow groundwater area levels to the 2024 drought. We're seeing a similar response. So we see a strong correlation between departure from normal rainfall to what's happening in our local shallow groundwater systems. So thinking about how we move forward with development of this plan. So who is this plan for? It's for all of us, right? So the collaborative is a voluntary organization. We can't tell anybody what to do, but we can develop recommendations for ourselves and also for our water utilities and jurisdictional partners who may be reviewing and updating their drought plans here in the next couple of years. So then thinking about what are the appropriate responses, what are the management actions, and what are maybe the longer-term policy actions or short-term policy triggers that could go into play when we have certain drought status levels. And then lastly, ultimately, how can we use this to inform steps towards recognizing an ecosystem's right to water through shared action? So I think that's really important for us to think about. We're putting so much effort into preserving and protecting and restoring. What happens with population growth? What happens with continued climate change? How are we going to guarantee water for these systems? Another piece of this is doing drought planning interviews. We've currently done over or around 20 stakeholder interviews. So thinking about how are people responding to drought? What are the tools that they have? What are their perspectives? So we're kind of in that listening phase. What we're hearing is there's a lot of data available. There's a lot of interest. However, there's limited communication about drought. So we're not even recognizing and validating what people are seeing and hearing. However, there's a sense of awareness and there's interest in taking action if we had those recommendations and framework. So what is needed is a local drought management approach tied to indicators, clear, unified, consistent drought messaging. And this may not be messaging that we're short on water supply for us as people, but how can we take action to protect our creeks and rivers? Cooperation, collaboration, it's a shared resource. Water is a shared resource. Messaging that cultivates connection, speaks to resilience, and then ultimately clear shared goals. So with that, I just wanted to give you a quick highlight. This is ongoing for the next couple of years. Maria Johnson is the coordinator and planning lead for the Santa Cruz Watershed Collaborative and who is conducting these interviews. If you are interested in participating, we'd love to have your perspective. Nadira, who's also in the audience, is working with Maria on those interview processes. So let us know what your thoughts, feedback is, and we're going to continue this for the next couple of years. So thank you.
Thoughts, questions? Coming in. Nope. And we'll get somebody before you. Oh, do you have, okay, Peter, go ahead.
Hey, Catlow, that was really interesting. When you're defining your local drought, levels and everything, there's about 80 different rain gauges in Pima County alone. And whenever you look at these rain gauges, and I've tried this at various sort of landscape levels, there's huge amounts of variation, even in a basin like Tucson. On the east side, you might be getting half an inch, in the northwest, nothing and things. So how fine of detail do you think you need in your drought planning, or are you just integrating it over what level?
Great question. So this map right here is kind of our early definition of what are our planning boundaries for the collaborative, not necessarily Pima County or other jurisdictions, but thinking about what are the planning areas that affect water supplies from a groundwater perspective, or shallow groundwater systems, or creeks and rivers. So thinking about that as a planning unit, then what's happening within that planning unit from a rainfall perspective, creek flow perspective, shallow groundwater perspective. So then thinking we could have different drought signals in each of these planning areas. And then there's different water providers that pull from one or more of these planning areas. So we brought in Klimas Matmiko to kind of look at and work with Pima County Flood Control District's alert system, who manages rainfall gauging stations as well as stream flow gauging stations, to look at what might be feasible from a technological perspective to then also may work from a planning area and response perspective. So we're still playing with that, and definitely the door's open for thoughts and ideas. Great question, thank you.
All right, yep. Go ahead.
Two things. First of all, there is a group out of Colorado called Coco Raz that where individuals are submitting rainfall data, and I think there's over 400 just in Pima County alone that can be viewed at on a daily basis. And secondly, my question to you is there any provision in the AMA when it concerns drought?
That is a great question, and I look at other AMA experts.
That's a very good angle. That's worth exploring, yeah.
Great question.
This is an active management area for folks who may not be familiar. There are active management areas that are designated throughout the state where groundwater withdrawals more or less need to be consistently monitored and reported on, which creates some different thresholds and different triggers, likely and potentially and ideally around drought. That's a great call. Thank you.
Hey, thanks for everything you have done to date for our water supply. My question is about rainfall normals, and this may have been addressed in an earlier lecture. I haven't been here for the entire time. But I think it's NOAA or maybe National Weather Service, they update the temperature normals. Is it every 10 years, every 20 years? I forget. So there's a new normal, quote unquote, every set amount of time, which I don't remember. Is there such a process for rainfall? If there is, I don't remember it, and I'm wondering how that factors in to your math on the topic.
Good question. So the U.S. Drought Monitor is a nationwide product that goes out that pulls in local rainfall data, temperature data, and then produces that drought index. So it's part of the standardized precipitation evaporation index, I think. So it ties in both temperature and rainfall. So then you can drill down to the state level, to the county level. I would have to put that question back to Clemus, to Matt Miko. He's been kind of our technical expert on how often they update those normals. Is it a 30-year running average or a 30-year average that gets updated every 10 years, I believe, but I may be off on that. Good question.
Oh, one more.
So one of the things that you mentioned in your presentation was about what do we do in the wet years, the really exciting and few, generally, wet years. And in California, they're looking a lot at, they're calling it managed aquifer recharge. It's using water spreading into areas, generally farms, taking more water and spreading it and applying more water than maybe they need to do incidental recharge, and then this incidental recharge feeds the aquifer for the next several years, maybe that, may be much drier. And it strikes me that there might be a few places where that could actually be done in the Tanque Verde Creek area. Wondering if you've been thinking about that too.
Yes, we have. Thank you, Julia, for that. We do have a separate reclamation planning grant that's focused on the shallow groundwater mountain front zone for both springs and shallow groundwater systems for enhanced recharge, demand reduction, as well as enhanced recharge to support those, especially from a climate resilience perspective moving forward. Those are longer term projects, so thinking less about short-term response. Doesn't mean there could be. I think the short-term response I'm seeing is there's opportunities with recycled water. How do we distribute that? When do we distribute that to buffer, especially during these dry times? And it may be, what I'm also seeing is a huge need for communication response. So making sure we build the culture of understanding and connecting to our current climate and not just waiting for the government to declare a drought, right? So what can we do as individuals, as neighbors, as HOAs, to even communities, larger communities? So yeah, we're currently in the brainstorming planning process, but starting with the focus on communications. And partly that's because we're in a extreme drought right now, and we are not hearing about it from a media perspective. We are not hearing it from a jurisdictional perspective. So the collaborative has started including drought information in each of its monthly bulletins. WMG has now just started to share drought information. So I encourage all of you, as we need to be communicating about this and then having those ideas bubble up for what are then the on the ground management to policy responses that can happen. So thank you for the question.
And thank you for the presentation, Catlow. Great job. Thank you. All right, and now we're going to watch, I can't believe it, but the last video. This one is called Ms. Beckers' Summer on the Santa Cruz with BIORETS. Following this video, we're going to take a break and we will resume. We're going to get trashed with 3 presentations about trash when we resume at 3:40. So here we go to the video, and then we'll see you after the break.
Hi students, welcome to 8th grade science. I'm so happy that you're part of our class this year. I want to introduce you to a very special place in Tucson that we will learn about in many of our lessons this year. If you've ever been to the base of a mountain or you've driven by the wash on the west and south side of the town, you've probably seen the Santa Cruz River. I spent my summer doing research on the river, and I want to share with you a little more about it. The Santa Cruz River used to be a real river. It flowed in different sections at different parts of the year. If you grew up in town, you probably know that now we only get a steady flow in the river if we're lucky when we have those big monsoons. But at a few points along the river, the city pumps in effluent water, which is reclaimed, recycled wastewater. This water will sink down through the river channel and replenish our aquifer underground, which is kind of like a big bowl of rocks below our feet that stores some of our drinking water. If this is confusing right now, don't worry. We'll be talking a lot about our aquifer this year. We got to visit the plant where they treat our wastewater this summer and learn more about how they turn that water that gets flushed away into something usable. Where the water is getting pumped into our river, we have new ecosystems created that look super different from our desert. And these are called riparian ecosystems, and they form along rivers. You can actually bring your friends or your family anytime to check out the river near 22nd Street and Mission Road. So our riparian areas host a bunch of super awesome creatures like dragonflies, frogs, turtles, snakes, and coyotes, as well as some wetland plants. Unfortunately, they can also be home to a lot of trash that floats into the river from the city. This summer, my group was looking at turtles in the river. So we were searching for a mysterious little dude called the Sonoran mud turtle, which is native to our river, but is very hard to find. We also found two other types of turtles, red-eared sliders and spiny softshell turtles, both of which are not native to the river, meaning that people brought them there, but they're not from here. And these guys can actually be bad for our mud turtle friends. So let's walk through a day in the life of a turtle researcher. We set out turtle traps at 6 different sites along the river, from downtown to Marana, and we wanted to learn what types of turtles we had and how many were at each site. So we also took measurements of the turtles, like their length and their width and their weight. Where our spiny softshell females were much bigger than the males, and they were super feisty, so they tried to bite us at least once a day. So our research days definitely could be super exciting. Sometimes we also caught fish, like catfish and frogs, like the bullfrog, in our traps. Everybody on the river wanted to try the canned tuna we baited the traps with. After measuring the turtles, then we measured characteristics also of the river, like how fast the water flows and how deep and wide the river was at those spots. We mostly found spiny softshell turtles, which like deeper, faster moving water. And then we found a few mud turtles who like murky, shallower water. And while we were on the river, we also got to meet lots of other great critters. Look at that coach whip snake in the tree. Water is so special and sacred in our desert, and this was a really great way to learn more about the importance of water in our city and how other animals and plants use that water. Hopefully this year we can keep learning about water together, and we can do a lot of other learning and exploring too. Thanks for watching.
If folks can make their way to their seats, we're going to start the first of 3 presentations on some very fundamentally important research here on the Santa Cruz River, and that's about trash. As we've discussed, we have had around $1 billion of infrastructure investment over the last 15 years to get clean water flowing in the Santa Cruz River, and yet there is a blight of trash in this river. So as you will be hearing over the next 3 presentations, there is active scientific research, data analysis, and forward-looking solutions on how we may be able to understand and fully quantify this blight of trash so that we can inform our policymakers, our elected officials, to continue in their good policy directions, in this case, in the direction of addressing trash. So first up, I'm going to bring up Nicole Richards and Andrew Fortman, both from the University of Arizona, who will present on assessing trash accumulation in the tributaries of the Santa Cruz River to inform mitigation strategies. Nick, Andrew, come on up. That's hot. Yes, you can clap. Woo!
What is up? I hope you're all ready to get down and dirty with trash, because that is what we're doing. Okay, to begin, who are we? As was said, I'm Nick Richards, they, she. I'm finishing up my second year of undergrad studying natural resources, conservation biology, as well as entomology. I have a very interesting relationship with Tucson. My family immigrated from Romania, but I still feel very connected to the nature in the desert. It is where I have grown up, even if it's not where my blood is from. And when I'm not doing academics, or I'll be usually hiking, playing D&D, crocheting, hanging out with my pets.
Hey, what's up, everybody? My name is Andrew Fortman, and I'm currently studying at the University of Arizona doing geography and urban development. And so being a part of this study has been so interesting, kind of seeing the intersection of our built environment, kind of how we consume, where our trash ends up, and also how it interacts with the wildlife that we see down at the Santa Cruz River. And so for our study, why do we care? Why are we looking at the trash that ends up in the Santa Cruz River? Now, first and foremost, it can't be understated the historical significance of this area. People have been living here for over 12,000 years, so it's really our responsibility to be caretakers of this history and be in a space that we're proud of and that is clean. And at the same time, we're lucky to have this green space. We have the loop going right through the Santa Cruz here in Tucson. And so, whether it's maybe your mental health walk of the day, or maybe you're taking photos of the wildlife there, we have so many reasons to use this space. And so we really want it to be clean, And, I guess we named it Harvey. I don't know if you can see the little praying mantis. We tried to invite him. Unfortunately, he was busy. But we're not the only ones that use this space. We also have to think about the wildlife, and they live there every day. So we should take care of it and have them, have a good home. And probably the coolest thing about this study is that we're actually adding on top of data that has already been collected. And so, as you can see right here with the map, we've already gone within the river the last few years and actually seen, okay, we know where the trash is, maybe there's counts and hot spots within the river. And so now it was our objective to go within the washes that kind of flow into the river and see if there's any connections, what their possible sources could be. And so when we kind of started our work, we had to identify, okay, where are the washes? You can see them, but you don't really know maybe where they are. And so luckily, Pima.gov, they have like a geographic information systems data that we can get. And so we were able to find all the washes within our study site, which was between Silver Lake Road to Grant Road. And so with this information, as you can see, all the dashed lines eventually run into the Santa Cruz River, which flows south to north, as you can see. And there were some challenges, as I'll kind of discuss later on, within being able to sample upstream and downstream, but this is kind of where we were in our study. And so when we kind of look at the method of how we did it, we would first identify the pivot points where the wash meets the river. And so once we identified that pivot point, we would then measure up 50 meters going downstream into the river and then upstream into the wash. And then once we kind of had that measured out, we would get kind of randomized points from 0 to 50, just go on our phone and get locations that we can kind of do our survey. And once we had those points, we would then create kind of a 10 by 10 square to kind of go out and survey and identify the trash that's within that area. And so this is kind of to put it in perspective of what kind of the field would look like. From overhead, this is the old Julian Wash, which is just south of Cushing Street, kind of where the streetcar goes over the bridge over the river. And so we would get down to the field, and you can see downstream on the photo to the right, that's going down into the river, and where we could do the 10 by 10 survey. But then we would go upstream, and there were some times where it would be infeasible, and we would have to adjust, which regardless, we were just looking for kind of the square area and what we would find in that space. And so some challenges, unfortunately, we do have people that end up living near the river or the washes. And so we really had to be self-aware and understand our positionality and just who we are. And so we never, if there were people living there, we respected their space. And so it's a very fluid environment. If people were there, we would have to come back, and yeah. So other than that, there was some vegetation issues. It was difficult. I don't know if y'all have ever walked through a haystack. I would not recommend it. But like right here, it looks like a grassy little area. I'm still finding birds in my pocket from the time we went out there. And at the same time, say if you ever go down to volunteer to do trash cleanups, there are issues where you will find needles. So it was definitely look at your feet, understand where you're stepping. Fortunately with that, you can identify with like orange tabs, you can see needles, but it is something to be aware of. And at the same time, weather is definitely a thing to consider. This is definitely not a study to do by yourself. So it's definitely that. And if you haven't been listening to anything I said during the slide and you're wondering if you're seeing double, no, those are two snakes right there. You know, that's actually at Old Julian Wash that we discovered them. So it was, you know, double snakes and double rainbows are kind of cool. They're both nice to look from afar, but the snakes are a little scary. But with that, I'll kind of hand it over to Nick, and then.
All right, yeah, so let's talk about some of those results from the study. Starting, we're going to just talk about counts, how many pieces of trash we saw. Now, we've divided this into one graph for the riverbed on your left, and then a washes and tributaries graph. Do pay attention, the y-axes are different, but that is just because there is more riverbed than there is wash. Therefore, we simply just had more sites from the riverbed, more totals. But we can still look at the proportions, which is the big thing we're trying to do here. And number one, across the board, everywhere you go, is food packaging. That is those clamshell takeout containers. That is a Reese's cup wrapper. It is a very wide category, but it is also just the most common. And generally, some other things we're seeing, top 5 in both, we're seeing appliances, clothing, liquor, and miscellaneous debris. And so these got out of order. Give me a sec. Next, we kind of see plastic bags. I'm just highlighting some trends that I thought were interesting that maybe aren't quite identical. There's way more proportionally plastic bags in the riverbed than the washes, number 2 in the river versus number 6. And kind of the hypotheses we have about that, plastic bags, also one of the biggest things we see, but they're extremely lightweight. Like, I think of all categories, they are one of the lightest. Very easy for them to just blow down or get carried down by water. And once you actually get into the riverbed, they're more likely to get embedded into plants, into sediment, that maybe they don't in the washes for some reason. And then we're also seeing almost the reverse of that, construction debris, which is wood pallets, bricks, any of that kind of stuff. Way more in the washes at ranking number 3, and just number 7 in the river. And part of that is construction debris, super heavy. Once it's somewhere, it's not going downstream. You need a lot of water to move even a single brick. But also something we just noticed as we're walking around the loop and different areas is that washes, because they kind of vein out into kind of urban space and the city, they're simply just going to be closer to construction and development than the riverbed itself. And kind of what is very fascinating, more nicotine in the washes, but we can talk about that later. Looking at weights. Generally, it's not surprising, heavy things are heavy. Appliances, automotive stuff, even clothing is very heavy proportionally. Just one trend that is interesting, we see more auto parts in the riverbed than the washes, even though they're extremely heavy. My personal theory for this is just that the river is wider than the washes. It's much easier for people to just dump in them. And that is also what I've heard from some classmates of mine who do research in a similar area. And just, again, weights and counts very different. If you had, right now, 10-pound toaster oven versus a Reese's cup wrapper, you'd be like, wow, toaster oven, way bigger deal. But there's way more Reese's wrappers out there than toaster ovens. you need, if you got one of the, why am I talking about Reese's? Because it was recently Easter, and if I see one more Reese's egg commercial, I'll lose it. You would need, from the moment that I started presenting to the end of this presentation, you would need to eat about 2 Reese's per second to equal a toaster oven worth of Reese's. And you might say, that's a lot of Reese's. We make about 25 million Reese's cups in the US per day. So just something to think about with just how abundant plastic is in our food packaging. Maps, as we saw, we have our previous hotspot map, and now we did not have time to make our own hotspots for the washes, but we have kind of this completion map, and kind of what we're seeing is hotspots that correlate with the washes directly that we can see is the Grant Road area, and there's about 3 kind of washes that are around there that correlate to the hotspot, and then the Arroyo Wash, which we were not able to get a ton of completion on, but we know that is physically one of the largest washes. It's taking in a lot of things from different parts of the cities, so it makes sense for that to be a hotspot. So again, we didn't have time to do any like t-tests or big statistical comparisons, but we now have the data and just that big data set to make that happen super easy. We have the proof. And of course, even though we have limitations of not getting all the full wash samples, not having that hotspot map, we are about to get those basically. So we really liked this picture from our most recent cleanup. Just kind of a reminder that conservation can feel like this huge uphill battle that is just so grueling and horrible, but when you have friends and community members to help you, it's way easier. And so we're hoping we can use this data to like inform where trash traps are gonna go, have some accountability. You know, we're gonna keep doing all these volunteer cleanups we usually do. And kind of now that we've done the riverbed, now that we've done the washes, we're looking at before-after surveys, seeing, you know, proving that trash traps and community cleanups have an impact. This is our current trash trap in Nogales. It's super effective, and we're hoping if we get some in Tucson, we can also start to take like samples out of those trash traps to say, hey, we used to see all of this in the river, now we don't see it anymore, and we see it in the trash trap. What's up with that?
So that kind of wraps up, but you're probably thinking, wow, that's a lot of trash, what can I do about it? So if you want, there's kind of QR codes right here to future events. It can't be understated the amount of events that Tucson Clean and Beautiful puts on. Personally, for me, it was about 2021. I gotta admit, I was kind of going through a low point. You know, with the COVID going on and everything, I needed to do something. And I'm a huge advocate for volunteer work. If you're coming across somebody that might be having a tough time, I highly suggest getting them out there and doing something. There's a powerful just result that you get when you're working towards something with other people. And at the same time, the Sonoran Institute, we are putting on events. They're pretty amazing. There's food, there's music. There's so many things going on that if y'all are interested, definitely check them out. But thank you.
Love to see it. Okay, questions. Trash questions. All right, we got 2. You can ask whatever kind of questions you want, I guess.
I'm wondering if there's any way, maybe from the data you already have or going forward with another survey, to identify potential point sources for the trash. Like, for example, do you think most of what's in the riverbed is washing in from the washes, no pun intended, or do you think it's maybe being introduced from road overpasses into the riverbed directly? And then related to that, any way to correlate possibly with point sources like the built environment, proximity to shopping centers or fast food places, that sort of thing?
So kind of that question of, is stuff really coming from the washes or is it coming from overhead? That is kind of the next big step we're hoping to look at. And again, if we had a trash trap and we see that it's not changing what's in the riverbed, then we can say riverbed's not feeding into it. Point sources, that's a kind of question. There's certain areas that we have been that are near certain brands of fast food chains and gas stations, and when we see that food packaging, you can tell who it's from. As far as what we can do about that, there's a lot of legal stuff with like, there's like a whole law of like the plastic bag ban in Arizona. You can't ban plastic bags, all that. But to answer that question, yes, sort of. We can't tell where every single piece comes from, but we can get some ideas.
thought I saw another hand. Was it the same question? Oh, Ernest.
Yes, I have two questions for you. The first one is, has there ever been a study on trash, the transport within a water column as to what you find at different depths within the channel?
So, great question. I do not know personally. I didn't do a lot of the background research for this study. We're kind of adding on. I will say something we do take into account on like our giant database is just floatability of average density. Like we know, okay, a plastic straw usually should float versus construction debris usually doesn't float. I think that is a very good question, especially just in the field. We've seen there's certain things that are way more likely to be embedded in the sediment, like clothing. That's almost always like been embedded. It's way deeper than other stuff. We've had cleanups where like there are people who were like, you know, don't spend the whole cleanup digging out giant tarp, and then they do it anyways. So I would say, I don't know if there's studies, but I would believe that is a thing. And what was the other question?
The second question is, do you do any testing for E. coli, especially in the washes where these, I guess they're homeless, they're not called homeless anymore, unhoused personnel, people live?
So, okay, the question was, have we done any testing for E. coli in the washes, especially where there are houseless people residing? The answer to that is, our study specifically, no, but we have also just, it happens that we usually wouldn't go if it's like too deep to wade through, but it just happens that when we were doing the surveys, almost all the washes were dry. I think that could be something interesting to look into. I know there are parts of the Santa Cruz where that is a concern, but that is not something we have tested for.
There is existing research on this, and it typically does find that most foreign substances, particularly where you're dealing with like saran wrap or plastic bags, things like that crinkle and fold and create micro zones within them, can harbor bacteria at a higher concentration than natural substrata. So it's likely that the trash that we're seeing in the river, that could be one of many environmental concerns regarding having trash, right? Then you're dealing with also a lot of these things that are floatable don't always, but tend to be petroleum-based. You're dealing with breakdown products from that, microplastics, but I don't want to steal too much from future presentations that we're going to have today.
Yeah, we do have a speaker coming up who is talking all about microbes, and all the tinies were the bigs.
All right, that's my job, and that's a wrap on these guys. Great job, you guys. Thank you. You guys did so good. Okay. Yeah, don't fall off the stage, please. So, next up we have Jacob Taylor, also from the University of Arizona. Jacob has been working alongside Nick and Andrew and myself and on this trash project. Jacob will be presenting on trash tracking transformed, enhancing environmental monitoring with data innovation. All yours.
Good afternoon, everybody. I'm Jacob Taylor, and we're going to be talking about, I changed the title, just practicing my alliteration, just flexing my muscles for y'all. Field findings, data infrastructure for the Santa Cruz River trash study. And I know this is every researcher's worst nightmare, talking about code, but we're just going to do a little bit, I promise. So just briefly, who am I? I'm Jacob Taylor. I'm a junior at the University of Arizona, and I'm studying computer science and environmental economics. And Sonora Institute is doing this trash study, which you just learned so much cool information about. And I bet you were wondering, man, those graphs look really cool. Where did they come from, and how did they do that? And that's what I'm going to be talking about today. But just a brief recap and kind of extending a little bit. Trash in the Santa Cruz River, pervasive, persistent, environmentally pernicious. It poses an immense threat to wildlife, water quality, and ecosystems. There's also an immense public health risk that comes along with it. And then, as well, Andrew and Nick were mentioning, of course, that really important loss of community cultural value. And interspersed throughout this presentation, I have pictures similar to how they had it, with pictures from our past trash cleanup, just to kind of give you some hope throughout, as we're going through all of these numbers of how much trash is in the river, that, yeah, we're doing something about it. And can confirm, like, this trash, kind of tough to look at, all picked up. It's gone now, which is amazing to see. Sonoran Institute has been categorizing, counting, and removing trash from the river for the past 3 years. And in this effort, over 22,000 square meters of the river has been cleaned, which I think is amazing. And that's spanned over 228 surveys and counting. And of course, with this, we have a lot of data, and that data must be analyzed. And so that's where I come in. On this building on success theme, we're looking to build on years of successful surveys. And so we're looking for that in a couple ways. We're wanting more effective data entry with data validation and just an easier way to do things. We're looking for more intuitive data storage, and we're looking for automatic analysis. And so we're looking at kind of what Nick and Andrew were looking at, that trash count and that trash weight. We're also obviously interested in floatability, which they touched on, and also namely recyclability. And the most important part here is that we want this analysis to be available as data are added. So let's say you do a survey, you enter it in, automatically you can see the updates reflected in the analysis. And so here's just a look at the old structure. We have this super ugly Excel file, as per usual. And this one Excel file handles entry, storage, analysis, sharing. It handles everything. And if you've ever used Excel, you know that this is kind of a bit of an issue. It makes it difficult to coordinate multiple editors. You have to do manual data entry, and namely, there's no data validation. And so this can be especially harmful when you have something like this that relies on delicate Excel formulas. And so all of these issues kind of feed into each other, where say you're doing some manual data entry, you enter a number that's not the right format, you enter a number in the wrong spot, very easy to do. Automatically the whole sheet breaks and none of your analysis makes sense anymore. You're scrambling to figure out where it is, and it's just something we're looking to avoid. So we can do better. And so here's our new structure. This is kind of a collaboration. This is kind of what we come up with. There's improvements to be made, but first we start off with Survey 123 for the data entry, and that's an ArcGIS tool. And this handles that data entry with data validation. So say you put in something wrong, it's in the wrong format, it's an unusual number. Survey 123 will yell at you. It'll be like, no, you can't do that. This is also smoother entry because it'll have like that Google Forms kind of feel where you're just slotting it in instead of having to find your exact cell in the Excel sheet. And then also that many contributors problem is dealt with smoothly because you have this one survey that holds everything, but multiple people can take the surveys at once. You just need the link to find it. And so right now we're doing data storage in CSV files, and these are automatically generated by Survey 123, which is another really nice thing. I hope that this kind of inspires you to maybe use this tool. Ideally, maybe in the future, we want to shift to a tool like SQL, if you're familiar, something similar to that, which is just kind of a more mainstream database tool, but this works for now. And we also want to be able to make optional updates to those CSV files from our Python script, which we're going to talk about now. We're doing the analysis in Python, and this analysis is automatic when new data is added, and our goal is to add up-to-date analyses to the website long-term. So now we have a quick little demo video, and this is just going to kind of take you through the whole process. So this is what Survey 123 looks like, kind of just a Google form, but it'll automatically record your date and time for you, which is super nice. And then also, this is just a beta version. Obviously, things might change. I'm just going to put my own name in for recorders, even though actually I've not gone out in the field. But Sonoran Institute uses the event ID as a way to identify different, like the temporal aspects. We're going to put one in there. We also have location description, which is just a brief description, obviously, of where we're at. And for this hypothetical, I'm going to say we're going to do Speedway and the freeway exit is kind of where we're going to be. And we're also interested in the type of site. riverbank or riverbed. We also have washes on that list now. And Survey 123 has this great location tool. And you can see I recorded this at the U of A main library. It'll automatically put that in for you, which is great if you're out in the field looking to do this. But let's say you're not out in the field, like in this video. You can also just navigate over, boom, there's Speedway and the freeway exit. Click, boom, done. Latitude and longitude will be recorded. And so you also have survey area size. We're gonna do 100 square meters as we talked about, that's pretty standard. And then we're gonna enter those numbers for the categories. And if you enter like a decimal point here, obviously that doesn't make sense. Survey 123 will say you can't do that. And so that kind of avoids that data validation problem that we were facing. Same thing, you can't enter like you can't enter anything with letters in it. It'll yell at you. So we're just gonna go through here, enter some dummy values. And obviously we would have more categories here, but this is just a short version for the video. So briefly, here is the entire survey filled out. We'll go down, we'll submit. Get that nice thank you. And so back here is what you would see on the back end of this. This is what Sonoran Institute will see. Reload the page. I think it takes a second, which is kind of annoying. But you can see there's that map up there, which is really cool. And then there, our response pops in. And just briefly scrolling through, obviously we have everything in there. Riverbed, survey area size. And we also have, there's that map, which is super helpful for that heat map that we were talking about earlier. This can be incredibly helpful there. Another important thing is that since this is linked to ArcGIS, when all of this, you'll notice there's that little analyze thing up there. Our data analysis for this project is slightly different, but that analyze can be used for things like heat maps, stuff like that. And here we're gonna export to CSV, and it'll automatically make us our little CSV file that we're interested in. Taking longer than I thought it did. And there it goes. Makes a quick little zip file, open it up. And you can ignore this for now. We'll come back here. And this is what pops up. And you can see complete response, CSV file complete. You can also see off to the right, we'll scroll over there in a second, that latitude and longitude is in there as X and Y, which is super useful for our ArcGIS needs. And so now we're gonna look at the whole database. This is what it looks like. Kind of a horror, I know, but this is a much more organized way to do things. There are zeros filled in everywhere. There's no incorrect data. And we also have these automatically generated columns like total items and total items per meter squared, automatically calculated. And now we also have another sheet, which I believe we switch to in a second, for the weights. And so this is automatically calculated by the Python file as well, using some multipliers that we'll show off in a sec, but this automatically calculates all of the weights. And it does that with this little dummy CSV. This is static, it doesn't move. These are just multipliers that say you have 10 plastic cups, you multiply it by that number right there, and it'll show you the weight. But now let's jump into actually making the graphs. This will be kind of like the back end of things. Obviously, when it's on the website, it'll look different. This is just like the behind the scenes effort. So there's a couple options. There's print, graph, chart, quit. But we'll do a print first, which basically just prints out the database. You can't really, it's not very useful for our purposes. You'd want to go in the CSV for that. It's just very basically to validate that everything's there. And then another thing to highlight here is that after every single operation, we have that little like successfully saved text at the bottom. And that basically signifies that every single time we're doing something, we're going back and checking, we're saying, hey, the CSV is still intact, right? Yes, it is. Everything is still exactly as it's supposed to be. We haven't changed anything. We're all good. So now let's get into the graphs. We're going to make that counts graph that Nick and Andrew showcased first. And we're not going to sort by a range of dates yet. We'll show that off in a sec. But it's as simple as saying counts, no, and there it is. And graph is automatically made. You can see 7,000 instances of food packaging, and it's automatically sorted in this descending order, even if, say, another item like liquor bottles, for example, moved all the way to the top, that would then become the top. It's always sorted in descending order. And you can automatically resize it like this to whatever size you want, and you just save it to directly to your computer. Now we have this weights graph, and the reason why it looks so wonky here is because it's actually in the same order as that counts, which is useful for the side-by-side analysis. So now we're going to do the weights. Highlighted, successfully saved. We're going to do the weights again, but we're going to show off a specific range. So I believe here we're going to do January 1st, 2024 to May 1st, 2024. And this is useful for these analyses, such as this is the tool that Nick and Andrew took advantage of for their instances with the washes, because their surveys were from in a specific time period and others were in another time period. Using these dates, we could filter them out and compare. And so here's the counts. It's in this weird ordering because the weights is sorted in descending order. And we can see for that date range, similar results, but appliances, over 700 pounds of appliances for those surveys. And so now we also have the pie chart option, which has a couple useful pie charts that we're interested in. We're interested in floatability and recyclability. And so we're gonna do floatable first. And you would just type this in, but obviously on the website, it'll just be a little button or even automatically up there. And so we'll hold off on the date sorting right now, but you can see, quick graph, 60.5% of trash is floatable. Same thing, you can stretch it out, and it just saves directly to your computer or instantly onto the website. And this is 3% is floatable by weight, which makes sense because appliances are heavy. And then we're just gonna do that one again real quick and just show off the comparison. So if we do floatable, and we're interested in a range of dates, and let's say we're gonna do January 1st to May 1st again. Again, this is useful for those repeat analyses where you're interested in comparisons. We can see that for this specific date range, 67% is actually floatable. So we want to go back and see, okay, what's different about these surveys? Why is there this increased rate of floatability? Where are they? Stuff like that. 4.7% by weight. And then we're going to do that on the other side of the scale as well. This will be from May 1st to December 31st of 2024. And in this instance, we actually see that there's actually a little bit less floatability, 53.6% by count, and then 4.5% by weight. And then the final thing we're interested in is recyclability, so we'll just make that for you real quick. You can see 16.3% is recyclable if you're looking at the counts. And then we got 11% by weights. And so basically all we're showing off here is just how easy it can be to make all these graphs using a tool like this. But now we can move on to the brief analysis. We'll keep it, how much time do I got? A few minutes, okay. We're gonna skip through the individual analysis and just get to the big picture then. Just our big picture lessons learned. We have that new analysis kind of extends and confirms the preliminary pieces of the study at a larger growing scale. As Nick and Andrew mentioned, the washes are pretty similar with some notable differences, but the washes are pretty similar to what we're actually seeing in the riverbed. But on a big picture, entire scale, 60% of the trash in the river is floatable. That means it's traveling far, it's very visible, and potentially harmful to wildlife. And also 16% of trash is recyclable, which means that it's avoidable, whether that be before it enters the river or even in some cases after it's picked up from the river. And just a reminder also, largest category by count is that food packaging, and by weight is appliances. And so we have some nice next steps here. We want to integrate this tool with the website, obviously, and we want more customizability. And so we have a lot of data, we have a lot of different types of data. And so we're looking to introduce things like vegetation index into the equation, like do certain levels of vegetation encroachment, catch different types of trash, things like that. And we're also doing, as mentioned before, the GIS analysis, more intense heat maps with this sort of data, which is already underway. But also with that data entry with Survey 123, we want to make this a widespread, community-driven data collection process. So we want citizen scientists, organizations, government agencies, anybody to be able to recreate this methodology that Nick and Andrew are talking about, and then easily enter their data to this database, and then automatically see the results. And so this project's data and analysis is useful for many stakeholders interested in point source identification, which will come later, but also just areas, hotspots, types of trash, such as Pima County, conservation groups, researchers and educators, and of course, the local community. Thank you.
Yeah, so for anybody in the room who has ever dealt with a medium to bulky Excel spreadsheet with 1 million tabs, like, this is the way. So very grateful for your talent and your work on this, Jacob. Question in the back.
Yes, thank you for that. It is fantastic. My question has to do with the bins, and so it actually might be a question for all of you presenting. It strikes me that if we're looking at putting hopefully more of the trash traps, the floating ones in the rivers and tributaries, that volume is maybe a really important factor thing to capture. And with the food waste category being so broad, is any of the data, like, retroactively, could we figure out that? Or, yeah, volume, like, since it's like food packaging is clamshells and, candy bar wrappers.
So yeah, in the current way that the survey is designed, food packaging is really broad. We don't have those several subsections. One option could be like going back and kind of seeing the distribution of them in the river, but obviously that might not be representative of what was in the past. But I don't know, Luke, are there any efforts to do this?
I think we could probably make some broad estimates on this, given that when we do these trash cleanup events, some of the partners will just like order a roll-off dumpster, and they know how many of them they go through, right? So a great utility of this study in general is we now have a pretty good handle on the makeup of trash in the river, right? So if in our efforts and when we do these trash cleanups, if we weigh every bag or if we get a roll-off, we can start to make some reasonable scientific assumptions about what the type of trash is collected within there. We know the volume of a roll-off. The other thing we can do is just put a wildlife camera on each of the trash traps. So when they start to get full, we don't have to sort of base it on a well-calculated but maybe more easily referred to solution in the form of a camera. Catherine.
Thank you. I have been told by stormwater people that it's a really good idea to bust the trash that's on my own property so that it doesn't go down into the stormwater drains and the washes and the rivers. For the citizen science effort part of it, would it be helpful for people who own homes to do that kind of data collection using 123 on their own property?
So yeah, I'd say we're most interested in the, like, if your property happens to include part of that wash or is actually in the river, something like that. I would say, yeah, or if you also live next to, like, near the river, but it's not necessarily your property, you can also just go down there, find a 10x10 plot, as they've said, and do your own stuff there. But yeah, I mean, if there is an instance where you have like that wash or that opportunity in the riverbed, for sure, definitely.
And there was an earlier question about, like, so far we've looked at, like, what Nick and Andrew presented on was 50 meters into each of the washes. We don't have a real sense of what the upper watershed for each of those washes are. The upper watershed of those washes are our yards and driveways and parking lots. So I think that as the study gets a bit more mature and we are able to expand this to a community science effort, that data collected, I mean, we tell people, the whole city drains ultimately into the Santa Cruz River. So when you pick up that blue and white styrofoam cup that's been rattling around at the bottom of your driveway for a week, you are preventing that from going into the Santa Cruz, or the breakdown products of it from going into the Santa Cruz. So there is utility in it, and I think there's going to be increasing scientific utility in it as we're able to expand the Survey 123 tool. And another thing that I think Jacob kindly understated here is that the way that we do this field survey, it's pencil and a paper and a clipboard. Then we enter it into Excel. Then we do the analyses, right? It's tried and true, but it's cumbersome, right? And it limits, like what Catherine's talking about there, it limits the usability of this to more people. So if people have an app and they can doink, here's my GPS location, here's how many, if you're on a plastic bag kick and you just want to do plastic bags, those are real data collected. And as we do those throughout the entirety of the watershed, it gives us a much better sense of what the overall trash load is in the upper part of the watershed, as well as within the washes and within the river, as we've been doing over the last couple of years. Okay, that was good. Let's do Ben first.
Thank you. I noticed what I thought was a significant difference in floatability for the different halves of the year. And I'm wondering if you had any insights as to what that might be explained. But thinking that between May and December is when you get your monsoon. And so maybe the stuff has already been flushed out, the floatable.
So yeah, I mean, I think you hit it right on the nail right on the head there. Like, that's definitely probably the best working hypothesis. Obviously, with our data collection methods, we don't exactly know, and so like we can't exactly say, yes, for sure, monsoons did this, but like best working theory, my personal working theory, probably monsoons are probably causing this, because similar like what we were just talking about, you have like that immense amount of debris like up on driveways, up on people's property, you know. In the monsoon season, it's much more likely to wash down, you know, whereas something like an appliance, that's not dictated by something like that. But yeah, from just this data, we can't yet say.
First, I love seeing code on the screen. Thank you. I guess I have two questions kind of related to the crowdsourcing aspect of the data collection. The first one is Luke just mentioned, if somebody just is on a plastic bag kick and they want to go count bags, but they don't want to count polar cups. Like, if the data field defaults to 0 instead of null there, like, you're not going to know that they saw polar cups, but they didn't log that. So that's one question, like, have you thought of that? And then my real question, though, like, more importantly, is looking retroactively, like, trying to measure the effectiveness of an intervention measure. So say we put a few of these trash traps in washes, and then we have, like, you could then, there's nothing to prevent you then from like drawing a virtual bounding box around an area in a riverbed and sampling the database at various points in time to see if that upstream trash collector is actually making a dent in count or weight. Is that doable?
Seems pretty doable. I mean, I'll defer to Luke on that specific question, but let me touch on the first one that you mentioned. Right now, the Survey 123 isn't set up to do that, but that was an idea that I had. If you noticed on like the CSVs, there was a column that said complete, which had ones in it, which is like, is it a complete survey? 1 for yes, 0 for no. And so a potential idea to expand that Survey 123 would be to like, okay, you have, if you didn't complete the survey, like say you're just interested in the plastic bags, you'd say no. And then there maybe if you say that, there's like a text box to describe what you did. And the other thing there would be, it's also kind of important to, in that way, separate some of those crowdsourced ones from the, like the Sonoran Institute specific ones, so that we can see like, just because the specific methodology is very sensitive, you know, and especially like if you're just on your property walking around, you're not, you know, taking a 10 by 10 area, you know, and so we want to have the ability to, we do want all that data, and it's all really important. We want to be able to see the total numbers. We also, for some of the data and for some of the analysis, we want to be able to separate those out.
And collectively, so Jacob has come to work with Sonoran Institute through the RISE program as part of the university's Arizona Institute of Resilience. And Nick and Andrew are with the EarthGrant program, also within AIR. I know some of the groups within here have taken advantage of that incredible program, and you're seeing in the last two presentations just what these student researchers are capable of doing, and that it is a tool that is available to all of us, and it helps launch careers. A lot of the folks who've gone through this program have now gone through grad school, have jobs, some federal, some non, some formerly federal. So it really does launch careers and provide some academic focus, some academic research. So I cannot speak highly enough about these three, and also about AIR and the EarthGrant and RISE programs. So they are at the end of the semester, so I don't think that the drawing the bounds analysis is going to happen, but hopefully we get another EarthGrant intern or RISE intern next year where we can take that on. Great job. Great job. Thanks, everybody. Good job. Very proud of you. All right, and our final scientific presentation of the day will be from Emily Waggoner. Emily is also coming to us from the University of Arizona, and will be presenting on plastics, pollutants, and microbial life in the Santa Cruz River. Emily.
Hello. Let me set my things down for a second. Okay, let's make sure that we're good. Beautiful. Thank you all for sticking around. The second day, end of day, is a tiring place to be. So I really appreciate you all hanging in there and being here. My name is Emily Waggoner. I'm a fifth-year PhD student at the University of Arizona. I'm coming from the Biochemistry, Molecular, and Cellular Biology department. Prior to my PhD, I was in environmental science and aquatic systems. So I've kind of integrated that past with now the work that I'm doing now, and those are kind of the lenses that I have been kind of coming together into this project that I've been working on this year. I am incredibly grateful to be here and to have had the opportunity to work in the Santa Cruz River, to learn from it and connect with it this past year. And I think we are now in this beautiful position. Ooh, let me move myself now that I think we're in this beautiful position to look at where the river is now and the health of the river and how we can continue to improve the health and cherish the river. And one of these challenges that we now, as Luke and Nick and Andrew and Jacob have highlighted, one of these challenges is the trash. So if you are looking on your far right in the upper picture, this is the trash that maybe we more commonly think of. These are the larger clothing, appliances, etc. These are things that you can see from a distance. But the trash and plastic problem or issue in the river, it extends beyond that. So as Luke has highlighted, as these plastics bake in the sun and sit in the river and flow in the river, they start to break down. And so then you start having these microplastics and all of these smaller fragments that are also a part of the trash in the river. And so you can kind of see, it almost makes like these little slurries. So you can see kind of in this picture here and down here, you have life in the river that is mixing with all of these little microplastics and plastic fragments that, create these little slurries. These two pictures are from Tumakakori National Historical Park, which we will be focusing on today. So one of the things that we can do, as a scientist, I see these challenges and I want to quantify it. And so for me, one of the ways in which we might be able to quantify trash and plastic in the river is through the chemicals that leach off of the plastic in the river. So I'm specifically looking at organophosphate esters, or OPEs, or one of my students, for instance, calls them OPEs. So I actively have to not say OPEs now. But these OPEs are a class of compounds. There's 30 plus of them out there. This is just an example of some of them. And these class of compounds, they're made by us. So they are human-made. You do not find them naturally. And originally we produced them to help in our plastic manufacturing and to resist flames. So we see them in all sorts of products now globally. This is in paints, lacquers, it's part of the automotive and plane industries. So they really are produced in a variety of industries that we now see globally. And something that I've been interested in my PhD is once they've been produced, they are then attached to products. And because they are added to a product, they can then be released off of these products quite easily and go into the environment. And so, where do they go? And should we be caring about where they go? The project that I was working on prior to this was looking at these OPEs out in our oceans. And we found OPEs in much bigger than detectable levels in the middle of the Atlantic Ocean. So they are all the way out in the middle of the Atlantic Ocean. They're somehow going from these manufacturing facilities or these products, they're being leached out into the environment in some way and ending up in the middle of the ocean. And so last year I started thinking about these results and thinking about here locally and our Santa Cruz River and started to wonder, could OPEs be found here in our Santa Cruz River? And if so, can this be a way for us to kind of take a different approach to quantifying the trash and the plastic in the river by identifying these compounds that might be leaching off of the plastic that we're finding? So with that, I will give this diagram its final debut for the day. It is a great diagram. So we are going to be zooming in on this black box here. This was the study site for us this past year. And we went out on a mission to see if we could find OPEs, and if so, where we might find them. I will walk you through this a little bit here. The sites that we selected, this was a culmination of so many people who have helped, who I will acknowledge at the end of the presentation. But we ultimately landed on 5 sites. Our first site was down in the Nogales Wash. Our second site was then at the outfall or downstream of the Nogales International Wastewater Treatment Facility. And then the final 3 sites that are really focusing on the trash dam that has been mentioned, and trying to see if we can get a look at these OPEs upstream of a large trash accumulation, at a trash accumulation, and just below the trash accumulation. And this is all, these 3 sites were at the, oh my gosh, excuse me. I know, sorry. At the Tumacacre National Historical Park. I apologize for that. Okay, and we took several types of samples here. For the sake of today, I am just going to be highlighting the OPEs, but we did the microplastics, we took sediment samples, so I'm happy to discuss those samples as well, but for today, it will be the OPEs. Just to give kind of a visual representation of the 5 sites, we have, going from left to right, we have the Nogales Wash. We have just downstream of, or at the outfall of the Nogales International Wastewater Treatment Facility. And then we have the remaining 3 sites upstream at the trash dam and downstream of the trash dam. And I will point out here the first amazing undergrad who has been part of this project, Paula Donahue. She was our outreach assistant this year and has just been phenomenal and is responsible for all of the pictures that we're seeing on this presentation. Okay, I'll walk you through very briefly the pipeline of, once we have a sample, how we go about measuring these OPEs. So we go out into the river and we collect a water sample. Once we get back into the lab, we can take this water sample and prepare it for several steps of filtration so that we have a small condensed sample that we can then analyze. Those steps are included here. So ultimately, we're doing solid phase extraction for any of the nerds out there. which is this stage here. What these preparations allow us to do is have a small concentrated sample that we can then measure by this instrument that you see here. This is gas chromatography mass spectrometry, which is essentially an instrument that allows us to identify which OPEs we're seeing and how much of each of them we are seeing. So with that, we spent the year preparing and planning, and there were several moments where my undergrads, both of them, who I'll highlight the second here in a sec, both of them came up to me at various points during the year and said, you know, I don't know if we want to find these OPEs or if we don't want to find them. And I think it really shows kind of the frame of reference that we were going into this, where we were curious to see what we would find. And I will highlight Kenan Prince, who is our research assistant. Kenan is responsible for so much of the research sample collection and processing over this past year. Okay, we have some preliminary results that I will walk through here. So I have kind of jerry-rigged these graphs a little bit to make it a little easier to follow the sites. So what we have on our horizontal axis are the sites. So site 1, 2, 3, 4, 5 going across. And what we're seeing going up is the amount of an OPE that we have. This is in picograms per microliter, which I will put into context here in a moment. This slide is for a singular OPE. This is the OPE we're looking at, trischloropropyl phosphate, or TCPP. TCPP, if we are moving over to the graph here, so we see some at site 1. This then spikes at site 2, and then it starts to go back down at sites 3, 4, and 5. So this OPE is very much so present here. And the pattern of this OPE, it seems to spike at site 2. Now this makes sense if we think about the uses of TCPP. So TCPP is used in, for instance, making our clothing. It's added to furniture, to the plastic bottles that we're drinking from. And these OPEs are not federally regulated. They are emerging contaminants. So we're still trying to understand their presence, what it means for them to be present, and how that influences the environment. And so the water that we use, eventually through the wastewater treatment plants, it makes some sense here to be seeing this spike. We see a second pattern that is also happening. So our instrument has the ability to measure 8 different OPEs, and in this sampling set, we detected 6. So there's these 2 patterns, the first pattern that we just saw, and this second pattern here. So now we are looking at a much lower amount. So before with TCPP, our maximum was 4,000. Picograms per microliter, and we're now looking at 40 as the max, so it is much lower. But now for EHDPP, for this OPE, we are now seeing 2 spikes. So we start at site 1 at the Nogales Wash. We go to site 2, we see this peak. It starts to go down as we get down to site 3, and we see a second peak right at the trash dam. Interestingly, EHDPP is made in food packaging. So there's some interesting things that we need to look into now more thoroughly now that we have these results. But again, this picture makes some sense with what we're seeing in these different sites. Okay, so OPEs are there. What does that mean? As I mentioned, OPEs are emerging contaminants. They are not federally regulated, so we are just now trying to understand what this means and if we should be caring about these levels. In Germany, for instance, they are regulated. So the German Environmental Agency guideline, just to kind of put what we're seeing into context, is 20 times higher than the maximum TCPP concentrations that we measured. So their cutoff is 80,000 picograms per microliter, much higher than what we have seen in this sample set. So not an immediate health concern. I am not gonna stand here and say we need to be looking at this for human health concerns immediately. What is maybe worth a further discussion and warrants further investigation is for the aquatic health. So in terms of the aquatic health, the concentrations that we measured are within ranges of studies where you start to see signs of stress in zebrafish. A lot of the research right now is focused on human health and using zebrafish as a model organism, which in some ways is kind of cool, because now we also get information about zebrafish. So it's within those ranges, and generally speaking, toxicology studies, this ranges from 2 to 10 times higher than what we saw in our samples. So all of this is to say that we do know that they're out there now, and there's a lot of curious questions to now be exploring in terms of the impacts that these compounds could be having out in the river. So what is next? There are so many questions that we could be exploring. For instance, how do these concentrations vary in seasons? Dry season, monsoon season. Are OPEs everywhere in the river? Are there specific extents that we should be focusing on? And really looking at that correlation between these OPE levels and the plastic and the trash in the river. And finally, our OPEs in other parts of the river. So I have not mentioned the microbes. If there's time, I will mention the microbes, but we've started to explore if these OPEs are interacting with the microbial community in the river. There are some microorganisms that are known to degrade these OPEs, so that's another thing that we're looking for in our sequencing data. Also, the sediments, the plants, the fish, everything around it. There are studies showing that OPEs interact with all of these different components. So there's a lot of work that could be done now that we know that they are out there. With that, I want to thank everyone. So everyone on this slide has played an integral role in this project happening. Every single person on this slide. So of course, Solange Duhamel and Christopher Frost, they're the lead professors on the project, and they were the ones that decided to go along with my crazy idea to go out in the river and see if we could find these compounds. Kenan Prince and Paula Donahue, who again have just been tremendous support over this past year. And then everyone else who in the community who has showed just tremendous amounts of kindness and willingness and openness to help and make this project possible. So Mike Medrano, Charlotte Hart, Tony Palmer, Luke Coles, Luke Cole, Nick Richards, and Ben Lumley. And finally, the funding for the project. So we were funded by the Sagarin Travel Grant. Paula, our outreach assistant, did so much work on stickers and brochures and going out and talking about this project. And the Sagarin Travel Grant largely funded that. And then finally, our project was funded by the Accelerate for Success grant at the University of Arizona. And so that we have to make sure to mention. And Allison Penn and Alba Falella at the Duhamel Lab, who came out and helped sample. And with that, I will take questions.
All right, we do have some time for questions. Yes. Right behind you, Catherine.
Yes, you mentioned on that first compound at the International Treatment Plant, It came out much higher, and you weren't surprised. I was. That's a water cleaning plant, so why were you not surprised?
Yeah, fair. So number one, we don't need to be looking for these compounds. Not very many people know about them, and they are just kind of starting to get out in the field. And so they're not regulated, and so nobody's really looking for them. So I think that's definitely part of it. And then the second reason for it is just because of the products that TCPP is used in. We are interacting with those compounds. I mean, they are in this room, very likely. So the products that we're seeing with TCPP would make sense to see this spike because we're using it in our day-to-day life, and then it's getting in the water and going from there. Exactly. Yeah, so one of the biggest sources of it, interestingly enough, is when we wash our clothes. The water that we're using will then show there are some studies showing that there are pretty high levels in the water from washing machines. Yeah, that's a good point.
Further questions? Well, all right. Emily, thank you so much. Great presentation. Well done. Okay, so as we have been doing for the last few years, we are going to be closing out Santa Cruz River Research Days with a poetry session. So Senora Josefina Cardenas was scheduled to read and present on this. Josefina was not able to make it today, but one of our great peers, a very talented poet, Logan Phillips, is going to be joining us over Zoom and will be assuming the role of Josefina, guiding us through some very beautiful poetry. And I'm holding the wrong sheet. So this is Santa Cruz River poetry, and Logan will give a little bit of a background on what we'll be hearing, who the poets are, and giving us a little bit of that grounding perspective around the Santa Cruz. Logan, are you there?
I hope so.
Oop, one more time. There he is.
One more time. Hello,
yeah,
hello.
we got you. Welcome.
All right. Giant face. Thanks, everybody. Thanks for having me. And my assistant Esme here. Just a small, I will not be attempting to take the place of Senora Josefina. I will tell you that because no one can do that. She is a unique and wonderful member of our community. But I do hope to share these poems. So hopefully you can see my screen there.
Yep, looks good.
All right, great. So just briefly to give a little bit of a context of the project here, this is a project that is a few years in at this point. It's called Sunset River Poetry, and it is put together by Favor Celestial, Senora Josefina Cardenas, Shirley Roman, and others involved in Favor, and generational elders, children. It's a really beautiful project, and it's supported by the University of Arizona Poetry Center, and they provide kind of facilitation. And what was unique about the 2024 edition is that the program moved every night. We were in various locations with various communities along the urban Tucson stretch of the river, including Las Milpitas Farm, the land of Chucchon, a mountain there near Mission Garden, inside Mission Garden, I believe, at one point, and in various places. And so it was very beautiful to all be sitting together, sharing that space, and to be writing together. I think this speaks to some of the pedagogy that Angel and Rebecca shared earlier, if I'm not mistaken, just being, forming those connections in place through creativity and community is absolutely a way to build consensus and community around protecting the river and creating the future that we all want to see. That's been an organizing principle at the Tucson Birthplace Open Space Coalition, or TBOSC, and it's wonderful to see that embodied so well in this project. So we're really grateful to have been included. And without further ado, Claire, have I missed any key elements? And if not, all right, awesome. All right, well, we're going to proceed with some poems. We want to share some poems. Esme is going to kick us off by reading her poem, La Coyote, which is right here. Oh, wait, no, wrong one, sorry. A Place of Refuge, a Home.
A place, a refuge, a home. A place, a refuge, with the city creeping, threatening to take over and make the place, the refuge, swallowed up by development. There it stands, on the brink, with animals living, thriving, along each foot. Plants bloom, coyotes, roadrunners, horned toads, snakes, people, and surprisingly, deep below lies the trash. Of how many people? 9 years worth of trash. Yet 15 feet up, an entire ecosystem lives all in the place, the refuge, the home.
It's poetry. Snaps for Esme. Good job. Nice. I'll share one that I wrote, and then we'll go back. We'll share some others from others in the community. Just for context, this one is titled 15 Julio 25 because this is day after, or maybe the day of, the hail hurricane. Day after.
Yes.
The hail hurricane that hit us here in downtown Tucson. And never have I seen anything like it. It stripped the plants to a degree that it actually smelled like fresh cut grass walking out into the garden afterwards. Um, so this is that poem. High, sky black, July snow, lime-sized and slicing, tiny cannonballs sluice and shred the plants as I watch, static hair standing to shock. Goodbye, dust. Goodbye, baby grapefruits and grapevine. So long, bird song, deafening, denting, car hood, popcorn. All this chaos, and still, all the sky gives is a blessing. That's that poem. And I do want to highlight the dedication to our First Nations here. We did meet also at the San Javier District Co-op Farm with our good comrade Amy Juan, and wrote there as well. And that was it was a real blessing to be with folks there and sharing food, which was an important part of this project. Of course, gathering in the shadow of the early sunset that we get on the Santa Cruz, thanks to the mountains, and sharing that space together. Esme, would you like to read Isidora's poem? We'll share a few more here.
Which one? Oh, that one. Okay. La Coyote. Her ears, a mountain range stretching far. Her snout is a river, even if it's unseeable. Her eyes are the deserts, wildlife very wise. Her neck is a hill that stands out and bends. La Coyote. The river. We pick up trash there. We ride our bikes through there. We dance there. We sing there. The river is a place of regrowth, love, happiness, sorrow. The river is a place of peace.
Shout out to Isadora. Regrowth, love, happiness, sorrow, peace. I love that. I'd like to share, speaking of love, I love by Senor Luis Salgado. The way my skin feels when you are approaching, the sounds you make that I feel only I can hear, the scent that triggers tears, as if to help fill you, this river, tus ojos, that the storming clouds replenish life, reassures me, that I would swim through mud like the fish, only to see and be a part of your flow here on this land. All I can think of is how beautiful it all was, and my hope that someday I can help bring it back. By Luis. And let's see, let's do a couple more here. The River by Miss Shirley Roman. My ancestors used the river for crops. My ancestors used the river to bathe. My ancestors used the river to fish. Today, my ancestors no longer live by the river, but still we are alive and well. Cry by Miss Rebecca Perez. Cry and cry and cry, cry every day to heal the river, to heal yourself, to heal your ancestors. Cry to move the water from you to the earth, to the ocean, to the sky, to your mother. Cry to teach your son to cry and heal, to release fear, to experience joy. Claire, can I put you on the spot to read your poem? Amazing. Thank you. And just to close, I'll share that I'm glad, Claire, that you mentioned not knowing what it was exactly and still jumping in. And this really is the intention with what is called social practice in poetics or social poetics, which is accessibility and a welcoming spirit for anyone who wants to be involved, sea inglés o en español o como sea. We really do welcome everyone to the river to experience it, not only in the data and the numbers that we absolutely need, but also in our senses and in our hearts and in our souls. And so with that, we want to invite everyone to our next poetry gathering, which is coming up this summer. We will be doing this project again July 14th through the 18th, starting at 5:30 p.m., which is just about the time it starts to cool off and we go into the night. And we will be moving in different locations again this summer, which I'm really happy to be experiencing. And in that spirit of inviting for folks who are interested in how poetry and place come together and building people power through the relationships created through people and place, we will be holding another iteration of Cultivating Culture, which is an ongoing collaboration between T-Bosque and the San Javier District Co-op Farm. This time it will be at the Co-op Farm on May 31st, starting right around 6 p.m. There will be food, there will be music, and some wonderful indigenous poetry, and you are welcome to come attend. And you don't have to write at that one. You can just come and enjoy some time at the farm. So again, that's May 31st. And we hope to write with you this summer. Thank you so much to the Research Days for making place for art, because art and science are two parts of a whole, and for everyone's wonderful work along this river that we love.
Weeks. So we will be sure to push those out. We also will do a proceedings from this as well that will share a lot of this same information. Sonoran Institute could not have done this without our lead sponsor, the Water Infrastructure Finance Authority of Arizona, with our many partners, with the gift of this space from Pima Community College. Absolutely, yes, Pima Community College, thank you. To our presenters, to the Tucson Bird Alliance, Friends of the Santa Cruz River, Tubac Nature Center, and to our steering committee who helped make this possible. A lot of you, especially the newer folks, really stuck your necks out to help make this program a reality. You all did an exceptional job, and I want to give my personal and Sonoran Institute's own thanks to you for everything you have done to make this a reality. I think with that, yeah, there's one more to go. Feedback. Your feedback seriously, legitimately shapes the way that Research Days comes together every single year. Venue, what did you like, what did you not like? Food, presentations, anything that really catches your eye or that you want to change, complement, boost up, please, please do this. We will use these data very thoughtfully to ensure that Research Days continues to be this really amazing event that we get to be a part of. From all of us at Sonoran Institute, to you all, to our language interpreters, to Christian for working, and I don't think sitting down for the whole last 2 days, and all of you, thank you. Have a lovely rest of your day. Please take whatever food you can and want to take, even if it means grabbing the whole chafing dish. This is the opposite of lunch rules. Go early and often and get as much of that food as you possibly can. Thank you, take care, and stick around if you'd like to. Bye, everybody.